Should we start our session, madam?
Yes, madam, you could start.
Okay, thank you, ma'am. Good morning to one and all. I'm your host, Vaishali Chaudhary, extending you a very warm welcome to the second day of International Conference on Life Science Research and its interface with Allied Sciences, organized by N.B. Mehta Science College, Bordi, Maharashtra. Yesterday, we had blessings from Honorable Vice Chancellor and Sir Dr. M.S. Gosavi, sir. We also had opportunity to listen to the highly enlightening talks from Dr. Richard Gomer and Dr. Hari Mishra. Today, we start with another eminent speaker, Dr. Ankur Patwardhan, sir. I request Ms. Jinnat Khan from Microbiology Department to introduce Dr. Patwardhan. Jinnat,
Thank you,
ma'am.
ma'am. I feel immense pleasure to talk about Dr. Ankur Patwardhan. Dr. Ankur Patwardhan is head at the Department of Biodiversity at Abasaheb Garware College, University of Pune. Sir has put everyone, Sir has put enormous work in his tenure of 24 years and yet counting. Sir is chairman of High-Level Monitoring Committee constituted by Ministry of Environment, Forest and Climate Change for eco-sensitive zone of Mahabaleshwar and Panchgini. Sir is also chairman of Board of Study at Savitribai Phule University. He is a member of Maharashtra State Biodiversity Board. Sir is actively serving as Honorary Secretary of Research and Action in Natural Wealth Administration, Pune. Being tirelessly active and true to his work, Sir has been awarded by Elsewhere Foundation and ISSC 3 Green and Sustainable Chemistry Challenge Award, second prize at Germany, May 2019. He got the grant for research projects which is supported by national and international agencies such as Department of Biotechnology, State Forest Department, Ministry of Environment, Forest and Climate Change, etc. His current research project focuses on recovery, including ex situ and bioprospecting of threatened plant taxa from Western Ghats of India. One of the 34 global biodiversity hotspots, his project on butterfly has been greatly appreciated in the media. Sir has witnessed various projects like industry-supported projects, international conservation agencies, national level agencies, local and university level bodies, development sector supported projects, etc. Sir has commendable hold over research related activity and organizes workshops, conferences, peer review of journals, papers, and environment awareness related activity. Sir has published article and paper in number of renowned journals and periodicals and has contributed to many conferences and symposia. He will speak on developing butterfly attractant for pollination and ecosystem risk. On behalf of everyone, I welcome you, sir.
Thank you, madam, for your nice introductory words. Am I audible?
Yes, sir.
Yes, sir, you're audible.
Okay. So, shall I start with my presentation?
Yes, sir.
Okay. I'll share the screen. Can you see the screen?
Yes, sir, we can see the screen.
Okay. So, thank you very much, madam, and I would like to thank the organizers of this conference for giving me an opportunity to share my experiences with you. Today, I'll be sharing my experience of working with butterflies. Yeah. So, ecosystem services are the benefits mankind accrues from the ecosystem. And there are various ecosystem services like pollination, biodiversity, and regulatory services like carbon sequestration, so on and so forth. And pollination is one of the most important significant ecosystem service. If we look at the last few decades, then we have witnessed a tremendous increase in agricultural crop which are pollinated by insects. And it is estimated that all over the globe, nearly 2 lakh species of insects, animals, they perform the service of pollination. And out of these 2 lakh species, more than 95% are insects. So by and large, 75% of the world's food crops are insect pollinated. More than 90% species of that grow in our forest, they are insect pollinated. So in nutshell, if we want to correlate with how it relates with the quality of life or human well-being, then we can say that every third bite of food that we eat is a product of pollination. So besides, we all know that we use honey in our day-to-day life, both as a source of medicine and as a nutrient as well. It is also a product of pollination, and there are many other natural products which are byproduct of pollination. Forest regeneration, we can say the seed will happen only when the insects will pollinate the plants. So the oxygen we breathe can be also linked to the product of pollination. So all these examples highlight the significance of insects or the pollinators with the human well-being. So over across the globe, various studies have been conducted documenting insect populations, and they have come up with alarming results. So here, if you can see the graph, on x-axis there are various insect orders, and on y-axis is the percentage decline. So more and more species, because of various environmental threats, they are pushed to threatened categories. Threatened species are pushed to endangered categories, and endangered species are pushed to endangered in the wild or extinct categories. So if we want to avoid this loss, then we need to take some urgent steps. So insects are nothing but a game of Jenga, where you can experience the bottom-up trophic cascade. If you remove the block, the entire ecosystem will come crashing down. So insects are, we can say, they are the building blocks of our ecosystem. And when it comes to pollination, though butterflies, though honeybees are the largest pollinating insects we know, butterflies are next to honeybees as pollinators, and they have been hardly explored for their pollination efficiency. So what we did is we first looked at the reasons of the insect decline, and we all know that there are various factors that are responsible for decline of insect population. The prominent one being the urbanization, the habitat loss, and the intensive agriculture. It has been also studied by various scientists, and the population of beneficial insects like butterflies in fields where the fields are spread with artificial chemical insecticides and pesticides. So in those fields, the population of beneficial insects such as butterflies has gone down drastically by more than 75% or so. So all these factors that negatively affecting the pollination process. The increased climate change has resulted in decreased time of flower opening, and very less opportunities are there for plant pollinator interaction. So what we need is to avoid this insect loss. So to avoid detrimental impacts because of synthetic insecticides and pesticides, we need safer chemicals. That means we need chemicals which are eco-friendly and which live in harmony with environment. So what we have done here is we are trying to develop uh, natural product based attractant for butterflies, which is eco friendly, which is water based, and which is specifically for butterflies. So, understanding plant pollinator interaction is a very key aspect in entire process, and the pollinator decides based on two cues. One is visual, and another one is olfactory. That which plant the insect is going to visit. So there is trial and error method. There is learning because the insects will visit the plants for the reward of nectar, and then they will decide whether they are going to visit the same plant again and again. So there are three learning processes like sampling by insects, then learning strategy, and then the decision making strategy. So excuse me. So what we did was we try to get insights from biology first. So by identifying plant species which are frequently visited by butterflies, and then we transform the biological understanding to chemical understanding in a way that those species which are frequently which frequently attract butterflies, we studied the chemical composition of various plant parts, including the nectar, and we try to simulate certain formulations which are natural product based, and to check their efficiency, the efficiency of the formulation on insects. So this project involves a combination of biology and chemistry both, and that's why I was awarded a prize by Elsevier Foundation in the Green Chemistry Challenge. So we primarily work in the Western Ghats, which we all know is a biodiversity hotspot, and butterflies are considered as next to honeybees as pollinators. And here we can see the overall richness of butterflies in our area. So India is very rich with more than 1,000 species of butterflies, and Western Ghats we support more than 330 species of butterflies, and many of them they are threatened species, and they also exhibit tight relation with particular plant species. So what we did, we shortlisted certain key butterfly rich sites in the Western Ghats and studied different plant species. And here you can see on x-axis there are different plant species, and on y-axis the number of butterflies which visit those plant species. So this gives us an idea of which are the preferred plants which are frequently visited by butterflies. So the data was recorded in such format, and then ultimately we narrowed down on to certain targets where the specific plants are visited by a maximum number of butterflies. Here you will see a particular plant known as ligustrum, which is very confined to the crestline of Western Ghats, and it supports more than 27 species of butterflies as pollinators. So as compared to bees, this is visited frequently by butterflies. Then there are different visitors of butterflies, which one can characterize into different families. So we also looked at the color preferences of these butterflies, and here you can see the different families of butterflies have different color choices. So color choices here, I mean that the color of the flower they prefer to visit. So we also studied their color preferences. Then the obvious question is why a particular butterfly visits a particular plant, and they mostly visit the particular flowers for nectar because nectar contains different sugars, and the sugars they are the energy source for these butterflies. So we studied the standing nectar crop that is available for butterflies, and we also profiled the sampled nectar using analytical techniques like TLC, HPLC, and we are also now moving ahead with the pollen morphology, which are butterflies carrying with them for pollination. So here these are the preliminary findings where we can see different species have different amount of nectar that is available for the pollinators. So then the moving to the second part, that is the chemical part of the study, was to look at the plant material or the species. Once we identify the species through biological studies, we subjected these plant material, the nectar samples, and the other plant material for through solvent extraction and essential oil extraction process, and then studied their efficacy on the butterflies. So here you can see that we have profiled different sugars from the nectar, and different species have different sugar composition, and the concentration of the sugar also varies with respect to different species. So our next target was to check their landing preferences, whether to check they get attracted to a specific color. So we have prepared artificial stations for them and recorded their landing incidences. So here is our experimental setup to check the landing preferences. So different color plates have been used, and the formulation which we have simulated, we sprayed it on the natural plants in the wild. So different color stations like white flowers, red flowers, and yellow flowers, they have been erected in the field. And in the right hand slide, you can see a butterfly visiting particular the artificial flowers. Then we spread these formulations on different plants and check the number of visits. So our idea was very simple. If the formulation is active, or it really attracts butterflies, then the number of visits, the frequency of visits should increase, and also the flower handling time. Because if the flower handling time increases, obviously it increases the probability of pollen getting attached to the body parts of the butterflies also increases. So we check the landing frequency and the flower handling time of these butterflies. So here we have checked this on a nectar feeder butterfly known as Tizay, and you can see the number of visits performed by tailed butterfly in 6 hours duration. So such several replicates have been done. Similarly, in the graph, you can also see the average flower handling time between the test that is unsprayed, test is sprayed with the formulation, and control is without any formulation. So we observed a clear-cut difference the response of butterflies to these formulations. Then there are certain butterflies which are fruit feeders. Not all the butterflies are nectar feeders. So we also did another bioassay on different fruit feeder butterflies like gray count or red spot duke, etc. And here you can see the seconds per visit. So the time spent by butterflies on the particular so this all indicates that if you have effective formulation, which is again a natural product based, which is eco-friendly, that will enhancing the butterfly visits to a particular flowers. So we also took this concept to the different farmers and training the farmers in raising plants which attract the butterflies. We also have developed a couple of butterfly gardens where we do the actual experimentation, and this also was covered largely by the international media. The efforts have been covered largely by them. Also the local media, and you can say even the social media as well. because this is the first study of its kind which combines the biology and chemistry aspect and utilizes green chemistry principle in designing of a formulation. So which is eco-friendly and which will enhance the efficiency of butterfly visits to particular flowers. So I would also like to share the one small video with you, and then give me a minute. Madam, can you see? Can you all see the video?
Yes, sir.
Yeah. Okay. So with this, I would like to end my talk over here. I am thankful to Elsevier Foundation and the Forest Department for their support, and also to Professor Anjali Kulkarni, Madam, Principal of Bordi College, for giving me an opportunity to share my experiences with you, and her entire team, which made this possible of relaying to the presentation to you. Thank you very much, and I'll be happy to take questions if any.
Thank you very much, sir. Anyone from the audience has got any questions? You're free to ask the questions. Yes, Suraj.
Hi, Ankur. Thank you for nice presentation. I have a curiosity-driven question. Like, what is the significance of colors on the butterfly? Like, if it is only for camouflage, I can understand it with respect to moths. They are not. They are more sessile and like docile, I can say. But butterflies are so active, and if they visit a white color flower, the camouflage anyway doesn't make sense, right? So can
Yeah,
you
but
comment on that? Thank you.
yes. So camouflage, as you rightly said, is primarily used as a escape strategy. But for the feeding purpose, they need to choose different flowers. And what we noticed till date is that different species of butterflies, or even for that matter, different families, they have a specific preference to different colors. So the reason why are we doing this, both in the field as well as also through artificial workstation, is to integrate such plants in the landscape. Okay, many a times we are posed with a question that how my campus will be more eco-friendly. In that case, if we have a careful integration of both the host plants and the butterfly nectar plants, which have a different flower color, then probably it will enhance the activity in our vicinity as. So they do show certain color preferences, and even in our artificial experiments where we have conducted artificial workstations, they do show certain preferences to specific colors. So both all the pollinators, in this case butterflies, they resort to both olfactory cues and the visual cues. So what we have observed is the color cues are primary cues. They will be used by the pollinating agent to come towards the flower, and once they are in the vicinity of the flower, it's the chemicals, the essential oils or the other chemicals which will actually direct them to a particular flower.
Thank you.
Uh, Sir, there is one question in the chat box from
Huh.
Vrushti Patel. She's
Huh.
asking, a common grass yellow butterfly is said to be a symbol of rebirth or happiness. Are they really related to such behavioral science?
Uh, no, it's not like that. But what you can say that these are the butterflies which appear first when the conducive season starts. For example, in our park, we observe characteristically two population peaks of butterflies. One, the dominant peak is during the monsoon, and the next somewhat subdued peak is during the spring season. So other than these two seasons, the butterflies, the population is very dwindling, or nearly you can experience a flat curve or flat line. What I mean to say. So during the month of April, May, no butterflies you will see flying around, hardly any grass yellows. But as soon as the conducive season arrives, say in monsoon, they are the first one to come out because they have tied over the dormancy period, and they are the first ones to fly out, and that could be the reason why they are known as symbol of birth. But that's the most logical reason I can say.
Okay, sir. Thank you. One more question. Butterflies prefer which kind of pollen grains? The question is from Zinnat, madam.
Okay. We have studied so far more than 50 pollen grains, their morphology using scanning electron micrography, and there has been no specific preference as such, but the preliminary findings, they are indicative that they, uh, I won't say prefer, but the pollens which are echinate, that is spiny, they are more their probability to get attached to the body parts. But there is no statistical difference between the types of pollens and the preference. Uh, all they have, there are triaperturate pollen, there are isopolar pollens, and there are many different categories of pollens which get stuck to the body parts of the butterflies.
One more question is there, sir.
Yeah.
Cross pollination is by chance methods. So can we treat butterfly in sense that they are able to perform sure pollination? This is a question from Vikas Joshi, sir.
Yeah, they do help in pollination, and the area where we are conducting our experiments is what you can say is largely devoid of other pollinators. So here you can definitely see say that when the seed set has happened, that certainly has because of the cross pollination, because we have cut off all other pollinators from the system. So yes, they are very much can be considered as cross pollinating agents.
Okay, anybody else? Anyone else is having any questions? Okay, Zinnat ma'am.
Thank you, sir.
Thank you,
On
madam.
behalf of all staff of N.B. Mehta Science College and entire fraternity of institution, I'm glad to express my vote of thanks. Thank you, sir, for honoring this conference with your great knowledge and experience with butterfly and enlightening us on importance of this entomophili for human being and importance of butterfly attractants.
Thank you, madam.
Thank you once again, sir.
Thank you.
After a colorful session with Dr. Patwardhan sir, our next invited speaker, our next invited speaker is Dr. Ratnakar Chitte sir. Now I request Mr. Vikas Joshi sir to introduce.
Hello, madam.
Yeah,
This sir is facing some problem.
okay.
Can we wait for 1 minute? I will call him, and if not possible, then we will start the session.
Yes, sir.
Just give me
Definitely
one
we'll
minute.
wait. We'll wait for 2 minutes. No problem.
Yeah.
It
Let me
is our
call.
play.
uh,
It
let me
was
let me call him and confirm. Right?
okay, sir. This was really a very colorful session by Dr. Ankur Patwardhan sir. It's a very fascinating word of butterflies. Now, whenever we will see the butterflies, we will feel and we'll try to understand how much of thought process is there, how much of chemistry is behind their behavior. Definitely, sir, it was very informative session for us. Um,
I wanted to know
Thank you,
whether the
madam.
butterflies. Hello.
Yes. Huh.
I wanted to know whether the butterflies um, have memory. Like, if they have visited that artificial flowers that you had erected, do they visit it again? Do they come back there?
Yes, yes. They do visit the same disk again and again. And if you change the sequence of the disk, then they are clever enough to recognize that and they will choose their preferred color. So such
I'm sure.
experiments have been done already.
I'm sure they might be feeling revengeful about being duped into coming there.
Yeah, yeah, but they do realize if you change the position of the disk, they do realize that the change has happened, and they do take some time to realize the change, but they notice it, and then they will again visit their preference colors disk.
So they are intelligent creatures.
Yeah, they are intelligent. Yeah,
Thank you.
yeah. Thank
Thank you,
you.
sir.
Thank
Um,
you.
Dr. Chitte has joined us. Now I request Vikas Joshi sir in charge of microbiology department to introduce Dr. Chitte sir.
Thank you, ma'am. Good morning to one and all, respected principal, madam, and my dear colleagues and invited speaker. Today, I am here to introduce Dr. Ratnakar Chitte, sir. Dr. Ratnakar Chitte, sir, has studied MSc Microbiology from North Maharashtra University, Jalgaon, did his PhD in microbiology in 2002 from Savitribai Phule University, Pune. He did postdoc in 2003 to 2006, supervised by Dr. William Ferry from University of Petersburg, US, BNL, New York. In 2006, Sir has worked as a research scientist at ARI Pune and then joined as a scientist C in 2010 to 2014. Sir has presented research work in several national and international conferences and awarded Young Scientist Travel Award from IUB UK 2000. Also awarded Young Scientist and Best Thesis of Year 2003 by Rotary Club Pune. Sir has published book chapter in Springer Publisher and 16 papers in national and international reputed journals. Sir having two patents published and is also reviewer and editorial board member of journals. His research area of interest industrially important macromolecule from extremophile. Sir currently working as assistant professor in UG and PG microbiology department at Vanita Vishram University, Surat. Sir, it's over to you. Thank you very much.
I think again he loses the connection.
He left.
He is facing some net problem. Let me wait for 1 minute, 1 more minute, and otherwise we'll start the video.
Okay.
Let's start with this video, I think.
Just madam, I message him. Just wait for a minute, half a minute. Right? Yeah, he gave the consent, so we can run the video.
Okay, sir.
Please run the video.
Okay.
Good morning, all of you. First, I like to thanks the आवाज Good morning, all of you. First, I like to thanks the Gokhale Institute Society and N.B. Mehta College, Bordi, for inviting me to deliver the talk in International Virtual Conference. I also like to thanks the principal and director of the college, as well as conference committee member for this talk. The today's talk is on role of extremophiles microbes in human life. So what is the role of extremophiles? We're going to see in this presentation. So first, what is extremophile? The extremophilic microbes which grow in extreme condition, and these microbes are having important role in human life, such as many microorganisms from extremophiles. They are producing novel biomolecules, and not only that, they are having characteristic of stability, because every biomolecule having stability at that extreme condition, then they will show the optimum activity. So this slide is explain the background of the extremophiles. Life in extreme environment, such as thermostability of molecule, having great role in industrial and pharmaceutical viewpoint. Because these micromolecules, once you identify, purify, then you can scale up this molecule for industrial point of view. We'll see in detail. And second reason, due to the global warming, the temperature rises in the earth. Due to that, there is life in dangerous. Even every life, starting from human to microbes, in dangers. They need to adopt the whatever increasing in temperature. Just like in extreme environment, the microorganism, they are tolerating high salt, high temperature, high pH, high alkalinity, high pressure. Likewise, they are also producing the enzymes or micromolecules which are thermostable and industrial point of view. So these are the some molecules which has origin from the extremophile. The DNA polymerase or top polymerase. The top polymerase is obtained from Bacillus stereothermophili, which is grow in extreme high condition. And the top polymerase is very useful in polymer change reaction, PCR. And this enzyme tolerated 95 to 97 degrees Celsius, and they show the optimum activity just like they have role in PCR. Second emerging area is biofuel technology. In biofuel technology, the extremophilic microorganisms which are producing lignocellulosic enzyme having great role to decompose waste agriculture material into the reducing sugar molecule. And this reducing sugar molecule you can use for secondary fermentation to the useful product. Either you can produce biogas, methane, or H2 gas, or ethanol, and nowadays gasoline, the ethanol and petrol composition used for biofuel technology. So these are the product originated from microorganisms which are useful for humankind or human sake. And you are hearing the environmental pollution caused by the vehicle bike, and this is the new generation turn into biofuel, biological fuel. So there is lot of work going on this area. Third is biomining. In biomining, you know about mining having acidic condition, anaerobic condition, and extremophiles are there. The acidic environment, they tolerating decomposing whatever, and the recovery of product you can get it from it. The certain microorganisms which are producing the carotenoid, and it is also used in biotechnological point of view. The extremophilic and their enzymes have extensively used, and their market is increasing, keep increasing day by day. The most significant and current lack of ability to produce most extremophile or extremogens on large scale required by industrial process. Once you discover the extremophilic microorganism, identify and optimize its production in lab scale as well as fermentation level at means maybe 2 liter, 5 liter, 10 liter, but industrial process optimization is bigger task. You can optimize the process parameter, media composition, everything, so that the process economic cost of industrial process will be cheaper, and the final product is the cost of the product is depending upon whole processing. So it will be adversely affect on the cost of the product. And the second emerging area is recombinant DNA technology. You can clone the genes encoding the enzymes or biomolecule into the certain vector, express it into the host system, either insect, bacterial system, or there are many hosts. You can transfer the recombinant DNA and produce the enzyme, either extracellular or intracellular, and then you can purify and get the yield. So these enzymes from extremophile recombinant enzyme used in food industry, pharmaceutical, agriculture, as well as in medical diagnostic kits. Extremophilic microbes help in plant growth promoter actually, and adaptation under harsh environmental temperature, salinity, pH, and drought stress condition. The extremophilic microbes used in agriculture also. You know the whatever the yield of the crop is decreasing due to use of chemical fertilizer, or the soil fertility decreases due to the use of chemical fertilizer. So if you want to improve the soil fertility, you can use the microorganism which is used as a biofertilizer, and it will improve the yield or nutrient content of the soil using microorganisms. The phosphate solubilizing bacteria as well as potassium solubilizing bacteria, then NPK, silicon solubilizing bacteria will improve the yield of the crop in the whatever particular land. As per their requirement, you use the microorganism so that the total soil nutrient will get increased, free soluble form, which are directly taken by the plant. Whatever the soil containing complex nutrient, it will not freely available to the plant, and plant growth get restricted. So here extremophilic, where the temperature is high in the soil due to the sunlight, you can use the extremophile and you can grow the crop in organic condition. So extremophiles, just like the soil having high salt concentration, many soils having salinity problem. Due to that, the yield will be decreases drastically. So if you use the halophiles, which grow at high salt concentration, they decrease the salinity of the soil and improve the yield. So I have worked on biofertilizer and pesticide for their production as well as isolation of new species for their application for agriculture use. So many microorganisms, they producing stress tolerating factors. Due to that, you can use this microorganism in different environmental conditions. The global market of extremophilic enzymes, which is used in different sectors like food industry, food and beverages, detergent, animal feed, biofuel, textile, pulse and paper, nutraceutical, personal care, cosmetic, as well as in healthcare and many more applications. These enzymes from extremophiles, they are having variety of application as per your requirement. So the protease and lipase are widely used for many industrial applications, and these protease have numerous applications in various fields. So I have worked on many thermophilic microorganisms for production of protease keratinase. In next slide, we can explain it. The bacteria rhodopsin is the pigment obtained from the halotolerant bacteria like Halobacterium soderium, which function light-driven proton pump. This bacteriocin having great role for the light generating mechanism. And similar kind of pigment is also you can observe in the eyes, which are used for improvement of many functions. So this is for medical application. Second area of extremophile is which are going to study by astrobiologists, the life in Mars, life in Jupiter, Moon, so that you can clue the origin of life from this planet. So many researchers are working on extremophilic microbes on the Earth. You can create the condition which are present in the Mars or Moon, so that you can study the biomolecule from them and clue the origin of life. I have already discussed that I have extensively studied the enzyme from thermophilic microorganism, which is isolated from the hot spring, and these are producing fibrolytic enzyme, cellulase, protease, keratinase, and lipase. And these enzymes are looked in industrial point of view. The keratinase having great role for degradation of hairs, nail, and it is also used for leather technology. Nowadays, keratinase having the role to produce amino acid from the waste hair, so that the amino acid cocktail you can use and commercialize for applied point of view. Second aspect is the enzymes like fibrolytic enzymes is produced from thermophilic streptomyces is also studied in detail. This enzyme is alkalophilic and work in dual temperature. Like it is stable in 55 degrees Celsius as well as its work optimally at 37. If it used for therapeutical purpose, it should work in normal temperature. So this is the application of the enzyme. And there are many enzymes which are used in industrial point of view, like cellulase is having role to decompose the cellulose from the waste plant biomass, and you can convert this biomass into reducing sugar. So reducing sugar, you can convert this reducing sugar into useful product using secondary fermentation. Either you can convert into alcohol or methane or H2 gas, depend upon if you want H2 gas, you can use aerobic microorganism, anaerobic microorganism. And for methane generation, you know the anaerobic methanogens, they have isolated from several sources used for biogas technology. And there are several microorganisms which are used for production of biodegradable plastic. You know very well, in environmental pollution, plastic creating pollution, you can avoid this undegraded chemical plastic. So you can substitute this plastic into biodegradable plastic. Several microorganisms produces the polymer, and this you can use as a microbial product for degradable plastic and use in various household. So there are many proteins which are used in food industry, pharmaceutical, beverages, and many applications. These proteins are used in diagnosis of the diagnostic kits. They are used in food pharmaceutical industry and many more applications. So there are a lot of alkaline proteins, metalloproteins, and many classes of proteins reported in different papers. So, but in the lab scale, primary level, you can put this research into the industrial level. And you know that if you invent some particular novel enzyme from extremophile, you can take the patent and protect your invention for applied point of view. You can contact the industry for further study, for collaborative work, just like scale-up process. In the lab, for example, if you discover some enzymes You can study their assay, identification, process optimization, all parameter, media optimization, then the purification method optimization. Then if it theoretical, you can study it in animal trial, preclinical trial, clinical trial, and then second step in that, the product scale up, production yield optimization. If wild enzyme for biomolecule is producing low, you have alternative source. You can use the recombinant DNA technology so that you can improve the yield of the product in the recombinant form. So you can clone the genes into particular vector system. There are lot of vectors are available like PET system, PT7, and lot more. So this is aspiration of the genes into the particular vector and transform into the host system where you can get micromolecule in extracellular or intracellular or periplasmic. If it produce extracellular, it is freely available in the cell-free broth. So you don't need to lyse the cell, or it is just require precipitation step or dialysis or furthermore purify you. You can purify it and use for application point of view. You can determine the yield and compare the yield from the wild bacterial system and recombinant. You can compare the activity wise also. You can compare fermentation, lab scale parameter, and you can report your novelty in that process. And third point, trials by safety trial, animal trial. You can take it, you can outsource it, and then you can build your product for applied uses. And this is very short talk because the time limitation. So this is the role of extremophilic microbes in human life, because microbes producing this, whatever I listed, only enzymes, micromolecule, antibiotic, certain drugs like L-asparagine and several micromolecule produced by this extremophile. They are anti-cancerous, anti-diabetic, anti-Alzheimer's, and many more. So you can use this micromolecule for applied use. So this is the short talk. Thank you, your cooperation. Thank you very much.
Yes, audience, if you have any doubt, any questions, then please ask. Sir is online right now.
Yeah, due to technical problem, actually it is run in hybrid mode. But if you have any question, you can ask me regarding extremophile, specifically from thermophiles.
Okay, sir, there is one question in the chat box. I will read out for you.
Yes.
What is the role of gamma protease enzyme?
Yeah, proteases are very specific in nature. They are used for many applications. Just like for vector designing, there are specific proteases are used for just like to cut down factor 8 site or something like that. So gamma protease, which cleave down specific amino acid, gamma amino acid in the peptide chain, we called as gamma protease. So they have specific action on the amino acid, on the particular peptide bond. There are alpha, beta, and gamma. So where they are acting, on that basis they are classified.
One more question is there, sir.
Okay.
Pyrococcus furiosus is supposed to have higher fidelity than Taq polymerase. So, other than PCR, what can be its industrial use? This is a question from Vrushti Patel.
So there are lot of top polymerase are discovered from many microorganisms. So the top polymerase action on its specificity as well as turnover number means in PCR the number of cycle how much amplification will get from the template DNA. On that basis you can use the top polymerase. The top polymerase origin is different from different microorganism. There are n number of top polymerase like platinum top polymerase which work very efficiently than the other. So just like your question is that, so it is from specific microorganism. So you can use depend upon your requirement, your efficiency of transformation from template DNA to the whatever n number of cycle amplification of the particular genes of desire. So you can use the selection of top polymerase. You can choose it according to that.
Anybody else is having any questions? One more question, sir, from Vrushti only. Uh, Hyperthermophiles breathe sulfur rather than oxygen. So, how we culture them?
So actually sulfur degrading bacteria I haven't studied. Specifically, I work on alkaline thermophile, but anaerobic condition, many thermophiles, means just like methane producer, they are also sulfur reducing, some of them, sulfur reducing bacteria. They are grow in anaerobic condition, means absence of oxygen. So they can reduce the sulfur and create the end product. So that is in short.
One more question is there, sir. Are all archaea extremophiles?
Mhm. Yeah, all archaea are extremophiles. This is correct because many archaea which are uh, their cell wall differentiation from normal bacteria, just like gram positive bacteria and archaea, their cell wall difference is there. You know very well basic thing because in gram positive bacteria we have N-acetyl muramic acid and peptidoglycan. But in case of archaea, the facial pseudomorine is there in their cell wall. So, and they are growing in extreme condition, just like methanogens are special class of archaea. They are growing in absence of oxygen. And second example is halophiles, archaea, which are growing very high salt concentration of salt. So that is the difference. All archaea are extremophiles.
Okay. Anyone else? Vikas, sir?
Okay. On behalf of N.B. Mehta Science College Bordi and entire team, I, Vikas Joshi, in charge of microbiology department, would like to thank the organizers of this conference for giving me an opportunity to share my experiences with you.
I think there is some connection problem with Vikas Joshi
Yes.
sir's. So, on behalf of NB Mehta Science College and the entire team of the
Am I
conference,
audible?
yes, sir, you are audible.
Yes.
Continue.
Hello. Uh, I thank our invited speaker, Dr. Aditya sir, for accepting our invitation and spending his valuable time attending and enlightening us on the external and their roles in human life. Thank you very much, sir.
Thank you. Thank you very much, all team.
Thank you, sir. Uh, the session of conference now we are going to have is a very special one. We always feel proud when our students excel in their lives. Watching them rule the world gives us a feeling of satisfaction and fulfillment. Our alumni have made us honored with their achievements. Their journey was never easy. What we see them today is a result of their hardship, passion, focus, and dedication. Today, some of them are going to share their experiences with us. The first person who is going to talk today is Dr. Suraj Warkhande. Suraj is a past student of Department of Biotechnology and is the first and the only student of N.B. Mehta to clear JNU entrance exam. He did his master's in marine biotechnology from Goa University and PhD from National Institute of Immunology, New Delhi. At present, he is a postdoctoral researcher at University of Salzburg, working on skin-resident T cells biology. Now, tomorrow's session, Suraj is also going to present his talk. So I'm not going to go in much details of his introduction, and I'll hand over the mic to Suraj to share his views. Suraj Warkhande.
Thank you, Vaishali, for kind introduction. Sorry, I will quickly share my screen. Can you see my screen?
No, Suraj, you're visible, not the screen.
Okay. Maybe I will try a window. Sorry for the delay.
No problem.
I will open my presentation in tab maybe. Sorry. I think I'm not able to share my screen for some reason.
No issues, Suraj. Can I play the recorded video?
Yeah.
Okay, no issues. We will be asking you some questions if anybody is having you. Be live with us.
Yes. Sorry for the...
No problem. Just 1 minute.
Hello everyone. Myself Suraj Warkhande. I am a postdoctoral research in IRIS Grad's lab in the Department of Biosciences, University of Salzburg in Austria, where we are currently doing basic research on human tissue resident T cells. the
I'm
presentation.
really glad that I got this
Just 1 minute.
opportunity of sharing my
1 minute.
experiences with you all. If I start from the beginning, I did my early education from a small village called Dapchari in Palghar district. And at that time, I already had intention of going into biological research field, but I did not know how. So I went. Hello everyone, myself Suraj Varphande. I am a postdoctoral research in Iris Gratz lab in the Department of Biosciences, University of Salzburg in Austria, where we are currently doing basic research on human tissue resident T cells. I am really glad that I got this opportunity of sharing my experiences with you all. If I start from the beginning, I did my early education from a small village called Dapchari in Palghar district. And at that time, I already had intention of going into biological research field, but I did not know how. So I went to a small science college in Chinchini for my higher secondary education, and unfortunately, I failed to clear the board exam. But I learned from my lesson that failure does not mean that you should stop. And finally, I cleared the board exam in my second attempt from Dahanu Science College. The term biotechnology was quite new and promising at that time, and I thought it was quite suitable according to my aims. So I continued my education in BSc Biotechnology College in N.B. Mehta Science College, Bordi. I feel very lucky to have started my higher education from Department of Biotechnology Bordi. Even though overall department was very young, their talent and the openness was very unique experience for me. And especially the faculty in the botany department of this college had really inspired me to read research article at that time, which really helped me in my research career. So I could say that my foundation of research career was laid in Bordi College. Here I would like to mention few names like Makaran, Vaishali and Anjali who really helped me to be more and more active with constant communication. Apart from education, they also gave me emotional as well as every possible help, and I'm really thankful to them. I'm really thankful to the Gokhale Education Society also for not only helping me but also recognizing my achievements throughout my career. After finishing bachelor's degree, clearing the JNU entrance for the MSc was supposed to be very difficult in Bordi, but I realized that most of the students did not clear it because they simply did not appear for the exam. So I went to the exam with little help from my friend and cleared the JNU entrance exam, where I got the opportunity to do my masters in marine biotechnology department of Goa University. Since the program of MSc of the Marine Biotechnology Department was of national level, I got opportunity to study with the students from different states of India, and I was benefited from their experience as well. The best part of the program was preparation of competitive exams together, due to which I cleared the Department of Biotechnology JRF exam as well as National Institute of Immunology PhD program. And finally, I opted for National Institute of Immunology PhD program, where I did my PhD on skin immunology. So during my PhD, I also further got opportunities to interact with students and faculties of different countries, which really benefited me for my research career. Was, apart from the research, I was also involved in extracurricular activities such as music, drama society, and I was also involved in community services, which really helped to build my CV for my future. But throughout my career, I have been in active communication with the body faculty, which I really enjoy and will continue to do so. During my student career, I went through a lot of things, and as an advice to my fellow students, I could say that there is really no alternative for hard work. So hard work is really important, and more importantly, the consistent hard work is very important. So keep working even if you fail, because failure are the real treasure of life, and one should really take care of their health. In long term, it is really important. And don't spend money on unnecessary things which can be useful on some very important occasions. And please grow up and take responsibility. This is especially for our Indian student, because I find it very concerning that Indian students are not independent really for long time, which really cost them significant time and money. So one should really be careful about these points, and one can find their own path. In India, we can use cricket anywhere. So, to convey my final point for the students, I would use this cricket example where all the cricketers in this picture have their unique way which makes them special in their way. So, please do not try to copy others in your future, but rather try to find your unique style which can help you build your career, and look at the others only as inspiration. And in the end, for my all fellow students, I would like to introduce you to an international community called Beyond Science Initiative (BSI), which we started when I was in PhD, and I am really proud of that we started such a great initiative. And please go online and join BSI. That will definitely provide you an international support which can help you build your career in future. And finally, I would like to say that never hesitate to ask for help. Thank you very much.
Thank you, Suresh, for and
Thank you, Suraj. Thank you for your inspirational speech. We have always been giving your example to our students, and we are looking forward for your lecture tomorrow. Thank you very
yes,
much.
thank you.
Next, we have Dr. Tejas Karadkar from Department of Biotechnology. Tejas is associated with Dr. Richard Gomer, whom we had privilege to listen yesterday. Tejas did his MSc in biotechnology from Mumbai University and also from Texas University. He completed his PhD in genetics from A&M University, Texas. And he has also won many awards, and he'll be speaking with us tomorrow, delivering his guest lecture. So today, now let's listen what Tejas has to tell our students. Tejas Karadkar.
Hello, everyone. Warm and best greetings to all of you. My name is Tejas Rajiv Karadkar, and I am an alumnus of this N.B. Mehta Science College, Bordi. It really feels great as one of the alumni from this institution, and I would like to thank Professor Dr. Anjali ma'am and Professor Dr. Chaudhary ma'am for giving me this opportunity to speak at this session. Remembering all the professors from my N.B. Mehta Science College, I would like to take a moment and I would like to take a moment and this opportunity to thank all my professors from biotechnology department like Pimputkar sir, S.P. Patil sir, Monica ma'am, Akansha ma'am, and the present staff of the biotechnology department. It was really fun during the botany lectures with Professor Kotiwar sir and Professor Gogri sir. And the some of the best lectures were from also zoology department. I had with Professor Kadam sir, Chauhan sir, and Sarode ma'am. And I can never forget with some of the interactions I had with Ghorude sir, with Nirgude sir, Suryavanshi sir, etc. So these were some of the professors that were really upfront for helping students at N.B. Mehta Science College. My plan or my path for educational trajectory was very simple, where I was planning to have an BSc, I was planning to have an MSc, find a spot for a PhD at an Indian institution, and be a scientist. But I guess there were some other things written in my fate, and I got fortunate enough to have this international experience. So after finishing my BSc from N.B. Mehta Science College, I finished my Master in Science and Biotechnology from C.K. Thakur College, Panvel. And that was another level of experience with more advanced experiments than the bachelor's level. But as a part of a curriculum from MSc, as a part of a curriculum for MSc, I had a chance to be a student intern in Associated Capsules Private Limited. And this was my first industrial experience. I would like to say that there were some experiments that used my knowledge and training that I've gained during my bachelor's and my master's. But at an industrial level, they have their own protocols and they have their own setup. They have their own requirements and the way they want to present results. So I would like to say that if you have someone from industrial background, or if you have someone from industry, please have discussions with them and ask them about the experiments they do, or ask them about the different approaches they have about the experiments at the industrial level. After finishing my MSc, as I was not able to clear some of the interviews after clearing some of the competitive exams for PhD spot in India, I started an institution for getting prepared for GRE and TOEFL, which are exams required for applying abroad, like in countries from America or United States of America, in Canada, in Australia, and Germany. These were some of my top choices. I was lucky enough to get into one of the institution as University of Texas at Dallas. And this is where I had to finish my master's again. This is something that I need to mention, that if you want to apply abroad, or if you want to apply internationally, you need to have a 4 years course of bachelor's. So that's why my 3 years course of bachelor's was not considered as a baccalaureate program. And that's why I had to finish 1 year of master's in UT Dallas. So this is something to remember. You can apply right away after your first master's part 1 to any abroad institutions, but you need to have at least 4 years of bachelor education from your parent country. UT Dallas was a very different experience. It was a very big culture change for me, a cultural shock for me. They have their own ways of classrooms. They have their own ways of examinations. They have their own ways of approaches and practicals. But in UTD, it was something that I learned about programming. Over here, I learned Perl language, or Perl computer language, which I guess right now it's a bit old dated, but it is still used for bioinformatics, for most of the programmings related to our genomic studies and everything. So UT Dallas was something that outside the wet lab, the actual experimental lab, I was more doing the research on a computer and Perl language. After finishing master's, I started a job in UT Southwestern Medical Center as a research assistant. And as a research assistant at such a big institution was a great opportunity for me. The main things I learned over here was the genetics, the molecular biology practicals first-hand basis. Like everything was given to me and I had to figure out the protocols. I had to manage and do the experiments. But the cherry on the cake was the microscopy department. It was a very big microscopy department, and I was learning confocal microscopy. I was learning laser scanning microscopy, and I was learning transmission electron microscopy at UT Southwestern Medical Centers. And these were some of the instrumental knowledge or technical knowledge that I was gaining during this phase of my path. Once I was happy enough that I have learned about the technical knowledge, I think I had to move on. And if you want some success, or if you want to explore more areas in the research, then you need to have a PhD as per the central dogma. And that's why I started applying for a PhD again at the in United States, and that's when I got accepted in one of the college in Texas A&M University. Now PhD was again completely different scenario, because I had a gap of 3 years. I took a job. Because I took a job, my motivation to learn something or to read a publication or to read a research paper was kind of at lower end. So I had to reignite that passion for my research. And that's where I met some of the great professors like Dr. Richard Gomer and Dr. Darrell Pilling. And they actually doomed me to read a paper. So if you, just as a tip, if you come across a research paper or a publication, you had to train yourself that just by looking at the figures, or just by looking at the data that they have presented, you need to get the gist of the papers. I do agree that it requires a lot of practice, but trust me, once you have that skill of understanding the paper just by looking at the figures, it is the most easiest thing to scan papers very quickly. So if you find a journal in library, please don't trade the entire thing. Try to look at the figures, try to look at the figure legends, and try to understand what the author is trying to say. And once you try to understand that, it's very easy, because it's always figure 1 is related to figure 2, figure 2 is related to figure 3, and that's how the paper goes. And every paper has its own skeleton. For my experience, say if it was a biology paper, we always had a target. Is that target expressed in the cells, where it is expressed, and in disease condition, how much it is expressed? If we mutate that target, or if we knocked out that target, then what are the consequences, or what are the results? If we have any inhibitors for that target, then what are the consequences? And to just figure out this questions, you need technologies, whether you need to do a DNA PCR, or whether you need to do an RNA sequencing, or you need to do an RT-PCR, you need to do an immunohistochemistry. These are all techniques, but asking the correct question is something very important after reading those figures, and that's what helps you to progress. PhD was a very great experience, I would say, and it really helped me in shaping my trajectory towards a very strict research-oriented profession. And after finishing my PhD, I moved to a company called Bio Inc., which is now focused on developing therapeutics on cancer and solid tumors and a disease condition called as amyloidosis. So I just want you to take a message from this trajectory, that if you look at my timeline, I started from biotechnology, which takes everything like microbiology, genetics, regular science, and plant biotechnology and everything, where I was more focused towards only molecular biology in UT Dallas. Then I was looking more into genetics and microscopy, gaining technical knowledge. And in PhD, I was exposed to a different area of human pathological conditions called as pulmonary fibrosis, which I'm presenting a talk that you can see in my coming session. And different set of instruments like flow cytometry, HPLC, and everything. And then I moved to oncology, which is cancer related. And now to just say these things, that what I have learned in PhD on pulmonary fibrosis is completely opposite of what I'm actually doing right now at Bio as a scientist. So my whole point is like, don't restrict yourself with certain boundaries, that if I'm from this background, I am only going to learn this much, or if I'm from this field, I'm only going to learn this much. Please have your interactions with your friends from other departments. Talk about chemistry, talk about physics, talk about botany and zoology. It's really important that you have this mixed knowledge, and that is what is going to help you. Like for my PhD experience, I had to learn chemistry, and I was learning chemistry again, which I missed out in my BSc. So it's really interaction. Do share your thoughts. Always ask questions. Like never hesitate to ask questions, relevant questions to the topics, relevant questions to the talks, and that's what is going to make you better. uh, I guess my 10 minutes are up, and there is so much to talk about. If you really want to have a conversation or want to set up a meeting, you're most welcome. I don't have my email ID over here, but you can definitely take my email ID from Professor Chaudhary ma'am. With that, I would like to again thank you, all of you. Please feel free to enjoy this session ahead, and I'm sure this would be a very great and very successful international conference. Thank you very much. I guess I will stop my recording.
Thank you, Tejas. It was really wonderful listening to you, and we are really waiting for your lecture tomorrow. Miss Bharti Maurya is a distinguished alumni. She completed BSc in chemistry during 2013 to 16 and then joined MSc Organic Chemistry. She stood first in MSc in the University of Mumbai and bagged 2 gold medals. She is our first student to receive university rank. Ms. Bharti is pursuing her PhD at Institute of Chemical Technology, Mumbai, under the guidance of Professor Bhalchandra Bhange. She has received prestigious DST Inspire Fellowship for PhD. Now let's listen what Bharti has to tell us. Bharti Maurya.
I think you're muted, Kirti. So there is no sound coming.
Sure. Sorry for the interruption.
Respected teachers and everyone who are present here, myself Bharti Rajkumar Maurya, and first of all I would like to say thank you to all of my teachers of chemistry department that they have given me this opportunity to share my some beautiful experience of my college, that is N.B. Mehta Science College of Bordi, which in front of you. So today I'll share some beautiful experience of my college during my BSc and MSc courses. So after completion of my 12th course, I had taken admission for BSc course in N.B. Mehta Science College of Bordi, where I took chemistry, biology and biology subject in my BSc course. So initially, I was not so much good in chemistry subject, and also I was not much knowledgeable about the mechanism of chemistry and their practical application. Since when I started to attend the lectures of the teachers of chemistry department, I got a chance to interact with well qualified teachers of my chemistry department, and also I found that the way of their teaching is really nice. That used to be like their way of teaching is used to be like that. The concepts would get delivered to students in a very easy way, and also they used to clear our doubts and deliver the knowledge not through the tough language of the books, but by giving a such example where from all students can relate and all are aware of it. So in this way, they used to interact with us, and also whenever we had any doubt regarding our subject, then not only in lectures but apart from the lecture timing also, we used to go to department office and used to interact with the teacher and used to discuss about the doubts and used to get it cleared. And also the practicals which used to be conducted during our courses, that used to be very interesting for us. We learned a lot from the practical courses also. So because all of these things, I became more focused towards my chemistry subject, and I started my interest towards the chemistry field, where I began to learn to understand the concepts of the chemical science field. So I took a chemistry course subject in my MSc course, where we had the four subject of the chemistry. It is organic, inorganic chemistry, or analytical and physical chemistry, and where I got a chance. I got a deeper exposure to the subject of the chemistry and the concept of the chemistry. And but I was very, my interest was very towards the organic chemistry only. So where I learned a lot from my respective teachers, and also in MSc there were seminars in every semester where we were given seminar topics where on which we had to give the presentation in front of the class. So these activities has contributed to build up the confidence level of our students. And also apart from this, our faculty member of the chemistry department has always encouraged us to participate in the extracurricular activities also, like any national level workshop or the competition where we could explore the concept more practical way. So these activities, contribute to build up the confidence level in students. So during my MSc course, I participate under the guidance of Dr. Patel sir. I participated in the one of the national level workshop which was held in the Guru Nanak College, where Dr. Patel sir and along with the two other students, including me, had participated in poster competition also, where we had secured a second rank. So that given me a much confidence level, and also build up my confidence level to get involved in such extracurricular activities. And hence I will definitely say that the teaching environment of our college, that is N.B. Mehta Science College of Bordi, is extremely nice, and also the faculty members are also fantastic, where any student without any hesitation can go interact with the teachers and also can discuss regarding any doubts and can get suggestions related to whether studies or the careers. And also the teachers has also supported us in that way. So, and also our college conducts the campus placements for final year students, where during my MSc course, the college had arranged a campus interview of one of the multinational company, that is Galaxy Surfactant Private Limited Company, where the three students in my department, got selected in the campus interview. Then after that, my MSc result that I have secured the first rank in the Mumbai University among all colleges, where I awarded 2 gold medals for securing first rank in the field of chemical science. So at that time, Dr. Kailas Sanap sir, who was my professor in BSc and MSc course in N.B. Mehta Science College of Bordi, has encouraged and supported me that I should get join in research area, where he has supported me, that he has just suggested me that I should apply for DST Inspire Fellowship, as I was eligible for applying fellowship, and because I had secured first rank in Mumbai University. So this is the DST Inspire Fellowship is one of the national level fellowship which is given to those students who are eligible and interested to pursue their PhD courses in better or good institutes. So I applied to this DST Inspire Fellowship in November 2019, where I qualified both the level of Inspire Fellowship under the support and guidance of Dr. Kailas Sanap sir, and also in the selection of institute and guide, Dr. Kailas Sanap sir has helped me a lot. So that I joined to Institute of Chemical Technology in Matunga under the guidance of Professor Bhalchandra Bhange. I would like to say thank you to Dr. Kailas Sanap sir, as well as all the faculty members of my chemistry department, my HOD, Dr. Nirgul Doctor, and my college principal, Dr. Anjali Kulkarni ma'am, and the whole college, that they have given me this opportunity, and they have provided me a particular pathway or direction to which I walked, and I took a first step in the research area. So now I would give a very short description about my research topic. That is, my research topic is the transition metal catalyzed carbonylation and double carbonylation reaction, the green method for the synthesis of the valuable chemicals, as the carbonylation is a reaction in very economical and novel alternative for the synthesis of carbonyl containing compounds like ketones, aldehyde, carboxylic acids, heterocyclic compound, dicarbonyl compounds. And these compounds have very wide application in the field of pharmaceutical industries, agrochemicals. Also, these act as a bioactive part of various bio-natural products also, which has various application as medicinal chemistry. So the traditional method or the conventional method from which these carbonyl compounds were used to synthesized was involving multi-step processes. Also involves those processes were having a low step economy, and also involving hazardous condition and very harsh condition. So, as a green alternative, this carbonylation is one of the very best, very good process. So here, this process for insertion of carbon monoxide molecule into a organic substrate. But as though it is a green technique, but it involves the use of direct CO gas, which is itself a hazard or very toxic gas for human health, and also very risky handling. So in order to avoid the use of CO gas, the CO surrogate has been used nowadays as an alternative pathway to CO gas, where these surrogates, CO surrogates, what it do, it liberates CO molecule inside to during reactions only, so that these surrogates, CO surrogates, are very safe, very safe and very non-toxic, also non-hazardous to human health, and easily handleable. So I'm working on development and the application of the geosurrogate chemistry in the field of carbonylation reaction, which may be catalyzed by homogeneous or heterogeneous catalyst. So, thank you to all of you.
Thank you, Bharti, and we wish you all the best for your future ventures. The next two students, past students, who are going to share their views are Dr. Rakesh Mayevanshi and Dr. Sachin Shinde. Rakesh, after doing his BSc in chemistry, he did masters in analytical chemistry and also advanced diploma in industrial safety. He obtained his PhD from Nayoga Institute of Technology, Japan, on 2D materials graphene synthesis, characterization, and application. Rakesh was Junior Research Fellow at Center for Environmental Science and Engineering, IIT, Mumbai. Rakesh was scientist at IIT and senior scientist at Netel India Limited, JNPT. Rakesh worked as project engineer at Ultratech Environmental Consultancy Lab. Currently, he is production technology development engineer at Toyota Gosai Corporation, Japan. And the other speaker is Dr. Sachin Shinde, who has done his bachelor's degree in chemistry, master's in inorganic chemistry, and PhD from Nagoya Institute of Technology, Japan. His PhD topic was synthesis of graphene and molybdenum sulfide and fabrication of their hybrid structures. Sachin has worked as project scientist at IIT Mumbai and senior research fellow with Ministry of New and Renewable Energy, Government of India. Currently, he is research associate at Cambridge Graphene Center, University of Cambridge, UK. He has 35 publications, one patent to his name, and he has presented research paper in various national and international conferences. I hand over this mic to Rakesh and Sachin.
Hello, all the teachers and respected students. And here we would like to continue with mentioning that during this journey, our teachers from BSc and undergrad, that they were quite helpful to motivate us on the learning curve that we are today. So in this environment where there is a lot of competition all around the world, we have to understand how to stand out and how to get into the stream where we can go ahead and start our career or get our career to a next level. So in this regard, many of our students, what we come from, like we come from the backgrounds which are not so much providing the ideal education or ideal conditions for the overall growth or overall going outside the India, because if you have the only learning of your subject matter, then there is a little bit gap that we face. So in this regard, we would like to mention here that knowing the English language is not the barrier. So you have to focus mainly on your studies. That subject expertise is very necessary. And even if you don't speak English, you don't have to get, think less of yourself and think about getting your career, getting yourself up to the mark and continue your journey. So from here, I will ask Dr. Sachin Shinde from Cambridge University. He has an excellent track record of academic track record. And I would ask him to guide our students from N.B. Mehta College that how to continue their journey and continue their progress in this regard. So please, Sachin, please take up. You can share the screen. Please switch on the mic. We can't hear you. Microphone is not on. Can you see my screen? Sachin, can you see my screen?
No, Sachin, we cannot see your screen. We can see your face.
Okay.
Rakesh, can you hear me now?
Yeah. Now I can hear you. So here's.
Yes, so I think some troublesome with my laptop. So is it okay? Okay, you share. I'll present from mobile.
You can see this screen.
Yeah.
Yeah, please go ahead.
Yeah, very well. Yeah. So first of all, thank you very much, ma'am, to introduce me and Rakesh as well. So as Rakesh, I would like to address few things before I start actually, because as Rakesh mentioned, it's not the most important factor over here which we want to focus is actually how students come out of that bubble and try to explore themselves outside of what they are doing at present and explore themselves somewhere else. So by keeping that in mind, me and Rakesh would like to address few things or few issues which can help them to go abroad and even try what they want to do and all. So you people must have got my introduction already, so I won't waste time on it. And so coming straight to the point. And one more thing, actually, I would like student to interact as much as possible, actually. So that will be better for us. So I want student to ask questions as well. So just feel free, because don't forget that whoever you are seeing now or presenting, these are the people who are sitting on the same benches where you are sitting now. There is not much difference. Only there is a time period gap because we have been like past that much time and we are here now. But otherwise, don't feel anything different. So it's just like friends. So just ask if you have any questions. Any questions, silly questions as well. It's fine. So okay, so why children and parents actually? Rakesh, just one slide back. So why children and parents consider studying abroad? Because not only in India, but most of the obviously you know that there are some countries where the education level is better than our country or even the other Asian countries. So everybody want to go abroad, and by this internet and everything, information and all, we get to know that what is good, what is not good, and even the track record of other people shows that okay, by going there they have did well, and so we can do the same. So by seeing this and listening these kind of things, everybody want to go abroad. But okay, that is okay. Everybody want to go abroad, understood. But it's not like as easy as to take a auto or cab from your college to railway station and all that. So there are few things we need to keep in mind in order to get to that. So as I mentioned there in the first slide, you can read that dreams aren't defined by social norms, but they are driven by passion. So this is not something big, some kind of big statement or something. This is actually very true. And why I'm saying this, because social norms means obviously there are many things which we feel that are holding us not going and all that. But actually, if you ask me, and as I mentioned, as Madam mentioned, and Rakesh also, that our background and all that, even I didn't know. Even for a few years back, I didn't know I'll be here it doesn't mean that I didn't plan my future. But the thing is, the key here is passion, hunger, and hard work. Rakesh, next slide. Yeah, so these are the few numbers. You can see these are the actually number of students applied abroad in the respective year. So if you see the year and the numbers, you can see they are increasing as year passes, except the COVID, where because of pandemic there were not many applications. But what it shows actually that yeah, people are getting more what you call interested, or more motivated to go to abroad day by day. So obviously the number will increase in the future as well. So Rakesh, next slide. So again, yeah, why children or parents consider studying abroad? So see, you need to understand the basic reason. It's not only that okay, we are fascinated by foreign culture and other things and all, whatnot. You people know more because we watch TV and all. So, but forget about them. Practically speaking, why? So India, we have certain kind of educational what do you call this procedure and the syllabus, how they teach us and all that. So we are mostly kind of what you call exam oriented. We have, and that is actually being from long time, and we have exams. So everywhere there are exams. But normally what happens? It's been in fade in our mind, and we just study for the exam. Most of the people must be studying at the last
This
moment.
makes our theoretical background, isn't it?
Yes. So exactly.
We have all the
we are
theories in our mind, and now we can jump after
yeah.
BSc. Yes.
So actually, because I have been in various countries, like actually I started in Japan, then I went to Korea. I spent 3 years in Korea, and then now here I already spent 3 years. So uh, why I'm telling this, because being in different countries, even for conferences, I go to, like, I have been to many countries, so I have interacted with other people. So the basic difference over here is, I know one thing, that Indians are known for their fundamental knowledge, their theoretical knowledge. They're very well known for that. So I'm telling you with proud that, okay, if you go abroad, nobody has that dare to ask you some fundamental questions, because they know that you will bring them down. So this is good.
Yes.
This is good.
That's
This
the
is
thing
good.
that we already have the power of the
This
theory.
is good, but yes, but what happens? These things actually kind of holding us back, because finally, like Rakesh is now in the industry and all that. So what matters in the future? Like when you spend enough time in the research and all that, what matters? Because see, theoretical knowledge can be gained in sitting in one room. If you provide internet and books and all that, you don't have to go anywhere. You sit in the room, close yourself, and just read and just read and just read and this. And as you grow up, as you become adult, your brain also get a bit improve when you read and all that. No? So capacity increases. So you can grasp anything when you grow old. So actually you don't need anything. But why? Why we go to the why the experiments are important, practical things are important, because those are the things only which actually bring you in the real application world. And so as I mentioned in the slides and all that, I'm not gonna read everything, because my intention was to make sure that okay, there are some differences. So later on, maybe ma'am can distribute this among students so they can read each and every point. I don't want to spend time on it.
Dr.
So there are
Sachin,
different.
can you tell our student the importance of English, or how much English is needed?
Yeah. So you see, English. Yeah, that is actually
Because when I was
Rakesh.
in BSc, when I was in I was a student, I wasn't able to speak in English, and that was the one thing that we think about ourselves, that we cannot do, we cannot go over there. So that's what
Yeah. Yeah.
I want our students to know.
Yeah. So this there are, as Rakesh mentioned, is, and actually most of the students, and like more than 80% students, I would say, and that is confidently, that they have that kind of what do you call that mindset in them, that okay, I can't speak, so I cannot go. So this is one of the most important factor which is holding you back by exposing yourself, because they won't know if you don't speak what you are and what you know. And then this is the same thing actually happening in India at present. But let me tell you that, like for Japan, Germany, there are few countries, and who are doing those people are very much proud of their language. So they don't come out of that, and they don't need to learn language. So Japan is so much advanced and all that, but you won't see Japanese people outside because they are proud of their language. They don't speak English, and if you ask them, they say they don't have to because they are good at it what they are doing, but they made themselves. So for language and all that, what you have to consider, what you have to keep in mind that see anything you want to learn and all that, you need to jump in first. You need to swim, you need to jump in the water. So it's same. So until you go into it and all that, it really doesn't matter. So just enter into that, and then you will, then flow will take you. So as Rakesh mentioned, language, forget about language. Language, उससे कोई फर्क नहीं पड़ता. Literally, when you go abroad and all that, one step ahead. When you introduce yourself on the international platform, then you will actually feel the gravity, and then you need to go through those, all the nervousness, all the problems. Your throat will get dry. Everything you have to go through it. Without that, you cannot. It's like it's must. So forget about that you will overnight you will learn English and all that. So you have to go through this. So that's a process. So Rakesh, next step.
In short, even if you don't know English, like डरने का नहीं.
Yeah, literally. But what happens? See, the actual thing is that English is a class in India. English is not education. Just remember this. English is a class. So when somebody speak in English, रास्ते में अगर किसी को देख रहे इंग्लिश में बात करते हुए, then you will look at that guy, and suddenly all of a sudden in 100 people you will notice that guy. Oh, he is speaking English. कुछ तो बात है इसमें यार. It doesn't matter. Come on, it really doesn't matter.
So अपना सब्जेक्ट का नॉलेज बढ़ाओ. पढ़ाई करने में केमिस्ट्री अगर पढ़ रहे हैं तो केमिस्ट्री आना चाहिए, बायोलॉजी पढ़ रहे हैं तो बायोलॉजी आना चाहिए, और फिजिक्स पढ़ रहे हैं तो फिजिक्स आना चाहिए. सामने वाला जो प्रोफेसर है उसको आता है, वो समझ जाएगा आपकी लैंग्वेज कौन सी भी हो.
Because see, language, English, Rapidex, you can use Rapidex and you can learn in 3 months. The chemistry, physics and all that you cannot learn in 3 months. You need to start from the background. You need to start from fundamental.
Yes.
And that's why you need to focus on fundamental. And that is taken care by our education system very well. Yeah. But what I'm talking about is practical and all that. So, Rakesh, next slide. So you see, now I'm kind of going in detail why. So this is all taken from internet. You'll get it. But I need to introduce in my perspective. So, okay. So first is more options than traditional courses. Rakesh, next slide. So one by one. So more options than plenty of courses, like 100 or 500 of courses and all that. So you have open freedom to choose whatever you need. Because in India, like, okay, मुझे ये पसंद है, मुझे चलो भाई किसी को screw लेके खोलने की आदत है, चलो भाई you need to repair something, you like it. Somebody like art, somebody like cooking and all that. So the best example is guys. Guys can cook. Yeah, guys can cook and they can do career in cooking. Because I'm a very good cook. My wife like my cooking very much. So forget about, you need to make one thing sure that nobody looking at you, and do your thing, and then only you can go ahead. Because see, me and Rakesh we know each other very well. We studied together. We did whole night together, study and all. We taught each other. So we know that our level of understanding, our level of how clever we are. But it's not like understudy, because there are many people who are much cleverer than us sitting or listening us now, but we are nobody. But if you ask me what I have experienced or what I have understood until now, there is only one thing. You can go, there are only two ways. One is brain and another one is hard work. So I don't have brain, but I can do hard work because that is in my hand. Brain to kabhi hereditary hota hai, or if you don't use it, then you won't get it. So that's a different thing. So, but I believed in one thing, that working hard is a choice. Brain is maybe or maybe not. It all depends. Mood ke upar depend hai. But if you work very hard, दुनिया का कोई माइकल nobody can stop you. Exactly. This is not something coming from internet or something. This is personal experience. Rakesh been in Japan, I was in Japan. So we have that kind of background, working 15 to 18 hours per day. Once I was literally didn't sleep for 3 days. I was working back to back because I built in myself. I showed, because as I mentioned, जैसे मैंने कहा, brain नहीं है तो वही काम करना है, तो कोई option नहीं है, survival है. So you have to do that. And I never like dream big, like very big. I have to be in Cambridge or something and all that. I kept working. Publications आते जाते हैं जब तुम काम करते हो. ग्रुप में काम करते हो, ऑलवेज बी पॉजिटिव, एंड दिस इज वेरी इंपॉर्टेंट. बी पॉजिटिव, वेरी पॉजिटिव, over positive. We have the term overconfident. I have the term over positive. So the best example I will tell you, the advantage of being positive is, so okay, so when somebody start a day in the morning, they go to the lab, they go to the work, something bad happens in the way, and then हमारा ऐसा होता है कि चलो पूरा दिन खराब चला गया यार, मतलब शुरुआत खराब हो गई तो पूरा दिन खराब चला गया. So this is wrong, because that is, you are tuning your mind accordingly. In my case, what I do normally, people take it as a being negative, but I take it as a over positive. So what I do when I start the day, I tell myself, worst things gonna happen in my life. Be ready for those things. So what happens? Anything happens in my life, like in the morning, something goes wrong. साइकिल का टायर पंक्चर हो गया, अरे चलो ठीक है, नथिंग वरी, मुझे पता था ये कुछ तो कुछ बुरा हो जाएगा, चलो चलो आगे चलो. So you go like that, think positive, everything will fall in place. So
you have to work hard and explore as much as possible.
या बट जब तक इंडिया में है हम लोग, जब तक हम लोग उस बैरियर में है, जब तक हमारे एनवायरमेंट में है, वी हैव टू फोकस ऑन एकेडमिक्स. लेट मी टेल यू दिस वेरी इंपॉर्टेंट थिंग, you need to focus on extra curricular and all that. It is after you achieve your academic goal, academic targets.
Sachin and Rakesh,
yes.
Sorry for the interruption. We are running up the time and we are having many publications.
Yes.
We have to wind up.
Yes.
So please,
please wind up last final.
Yeah.
Which one I will? Yes. Okay.
No, just go. I will just read it in 2 minutes. It will not won't take time.
Okay.
Just go back. Okay. So go fast. So there are some bullet point I have mentioned here. And then accessibility of admission, more option to traditional. As I mentioned, traditional courses, there are plenty of traditional courses in different countries and all that. Everything is available online. You can just read out, and out of that you can find your own interest, and then you can go accordingly. Immigration, settling and all that, outside and all that, you don't have to worry about it. Once you start, you will go through that, and then you will reach nowhere, because it's not difficult once you enter. The research opportunities, obviously, facilities are better in other countries. So obviously, you can go much faster, and whatever you want to do, you can implement straight away. Better career growth, obviously, again, since you have better experimental hands-on and better research experience, obviously your career will boost. Next, Rakesh. So this is it's just holistic experience. It's not just an education, because it's not only education. There are many things like learning the culture, other cultures and all that, language, and different kind of people from different countries. So sharing their experiences. So sharing somebody's experiences always saves your time. So you don't have to go through your own. So, meeting more people, sharing their experience helps you a lot. Brings you international exposure and all. Okay, left. Next. So advantage of studying abroad, this is different perspective, that is different perspective. So again, advanced learning techniques, obviously, because of, as I mentioned, better job opportunity, chance of interactive. I mentioned all this, financial learning curve. पैसा आ जाता है once you have enough experience and research from good institutes and all that, money will come after you. It allows you to experience a difficult style of education everywhere. Actually, most not all, but I'm talking about some countries where education is much better than India. When you go there, you will learn new and difficult things, which will make you more mature. Next. Yeah. So go next. So these are the problems you can face when you go abroad. So choosing the course and completing the campus application process and all this. So this is very important. Since there are plenty of courses, you need to focus on what you want to do, and everything is available, and just choose that. So it's a very simple part. Don't think that, okay, I want to do that, but it's not available. Everything is available. Financial worries, there are different kind of scholarships and all that. Study loans and all that you can take actually, and all everything. You will have the guidance. There are people sitting for that, for guidance, which will provide you guidance and all. Next. Cross-culture barriers. This is also important, because since you are coming out of your bubbles and all that, you'll meet new people, everything will be new, but it's for the moment. After some time, you'll be one of them. So don't worry about that. Okay. Issue related to homesickness and integration. Obviously, this is very common. It remains for 1 to 3 months. But after that, you'll forget your family, and you'll even forget to call them. Why is your student profile important? This is important. So this is very important. I'll spend like 2-3 minutes on this. why is your student profile important when applying to top universities? So see, when you are applying for somewhere, for something and all that, they don't know you, they don't know your background, they don't know what you are and all that. So it's very important to make your CV, make your curriculum with and all your resume and all that. So you need to really focus on that. Do extracurricular activities, not only educational. Educational grades are very important because that's what is gives you your first impression. But other than that, when you go abroad, they need other what extra factor you have, what is X factor you have. So you need to do other than that. एजुकेशन तो कर ही लोगे, पास तो हो ही जाओगे, उसके अलावा तुम बाकी क्या करते हो? प्रेजेंटेशन किया है कहीं पे, या you have some social things, you help somebody and all that. Everything put in that. That helps you a lot. Next. So yeah, beyond classroom and academics, as I mentioned, demand for scholarship, stand out from the crowd, be unique. You have to do something because see, whole class has 100 people, everybody study for the same syllabus, but only few can succeed. And the reason is their approach is different. So come out of the box, have some different approach, then you will go ahead. Next. Next. Last. Back. So this back. So these are back. Some countries in some countries and some universities in some countries, which are most of them are actually cheap and some of them are free. So these are I listed the name. If somebody want to approach, they can go through this list. Next. So this is actually the survey and all that who says that with respect to US. So when somebody come study from US and all that, okay, so they have developed ability to go along with people and all that 91%. If you see the numbers and all that, so everywhere is above 89, 90% and all that. So this is actually working when you go abroad. Next. Overall agreement that US degree is valuable in the country that I am living in. So 85% people says that degree earned outside is much more valuable than earned in your own country. And this is other countries. Saying that earning a degree in the US was worth the cost. Obviously, money-wise, obviously you will earn more when you have a foreign degree. Okay. Employment status by top citizens. So after getting a degree outside, you have more chances to get a job in your own country. And then, Rakesh, if you want to share, I'm done.
No, thank you. Thank you very much for Dr. Sachin for sharing with us.
Dr. Sachin, what you can do is you share your PPT with me. I will put it in group, exclusive group, so people. It will be helpful.
Sure.
Okay, thank you.
Sure. I have made one PPT also. So how to come to Japan and what are the constraints and how would you able to apply for scholarships? What will the lifestyle? I will share my thoughts and I will share my email with you. So any student who want to contact me, you can contact. Thank you very much.
Thank you.
Thank you. That was very nice of you, Rakesh and Sachin. We end with our morning session over here, and we are starting with our presentation. Sorry, Roop Malik sir, that we are late by 15 minutes. I'm extremely sorry for that. Those who are in session B, they should leave this meeting and join the links given to you for the session B, and those who are in session A will continue with this meeting. I hand over the mic to Vrishti ma'am to conduct this session. Thank you.
Thank you very much.
Yeah, before Vrishti starts, I would like to say something. Today we are having many papers. So I will request all the participants to restrict yourself to 8 to 10 minutes if possible, so that we can finish up in time. Thank you very much.
Thank you, sir. Good afternoon, everybody. Myself, Vrishti Patel, and we are here to begin our session A for paper presentation. So we are fortunate enough to have Dr. Roop Malik sir as a chairperson for today's session. Welcome, sir. Dr. Roop Malik sir is an Indian biophysicist who works on nanoscale molecular motor proteins that transport materials such as viruses, mitochondria, endosomes, etc. inside living cells. His lab focuses to understand the function of molecular motor proteins and its role in pathogen degradation and lipid homeostasis in liver. Sir has completed his PhD with over 20 international articles. Currently, Dr. Roop Malik sir is a professor of biosciences and bioengineering at IIT Bombay. He has been awarded G.D. Birla Award, Infosys Prize 2018, Shanti Swarup Bhatnagar Prize for Science and Technology 2014. I welcome Sir and I request him to begin the session.
Thank you, Rushdi. Can you hear me?
Yes, yes, sir.
Yeah, okay, great. So thank you for giving me this opportunity. It has been great to attend some of the talks over the last 2 days. I think there's some voice in the background. Yeah, they are muted. So we will get started right away. And we will get started with just 1 minute.
Shall I call out the names, sir, for presentation?
No, I have the names here. Yeah, so we start with Mr. Ramdas Bhong, right?
Yes, sir.
Yes, sir.
Yeah, so please, Mr. Bhong, who will tell us about the hypersaline environment in the solar saltans of Mulund. Please share your screen and let's get started, and we will time you for 8 minutes, Mr. Bhong.
Yeah.
Sorry to interrupt, sir.
Yeah,
but let me inform all the participants. Please note that I will have a 8-minute reminder call, and after that, within 2 minutes, you will have to wind up your session.
Yeah, we will keep to time very strictly.
Yes, I will interrupt at 8 minutes, and then you will have to wind up within 2 minutes.
Okay.
Yeah, thank you very much. Shall I present my screen?
Yeah, please share your screen and let's start.
Yeah, please. So, I hope my screen is visible to you.
Yes, you can start.
Yeah, thank you very much to provide me our opportunity to present my paper in this international conference. And I request all of you be safe for the COVID situations. Thank you very much. So I have presented this paper of physiochemical analysis of water sample collected from solar center of Mulund as a preliminary study for my PhD thesis, because I am working on the isolation of carotenoid producing archaea and bacteria from solar sultan of Mulund for the carotenoid production. So most of the microorganisms are nowadays isolated from soils, waters, and especially from oceans. So the archaea and halobacteria having nature is a salty nature, and they are mostly found in a high saline water. And that's why I used to select the Mulund as a seasonal salt plant, and it is a traditional in the west side of Maharashtra, India. So why I choose the carotenoid? Because carotenoid having the medicinal properties, having the carcinogenic properties, because most of the cancer diseases are cured using the carotenoids, and even it is used for the heart disease and for the therapeutic development products and anti-inflammatory actions also. So we'll go for the materials and methods. Mostly the solar saltans are the 7-pond system. That is, these are the interconnected ponds with each other. The start with the pond 1, 2 and end with the pond 7. Mostly you call as a high crystallized pond of the seventh pond. In that, we have collected the samples mostly in the summer season, because in that region mostly the high salinity waters will be observed, because in winter seasons or in rainy seasons it will be more challenging. So I selected in the May month for the selection of the samples, and at the sample selection sites we have collected samples in the glass bottles and all. At the same time, we measure the temperature for each ponds, and we have collected separate pond have a separate samples. Temperature and pH at the time of the collection were measured. Even we go for the different physiochemical parameters like total solid, total dissolved solids, total suspended solids, pH and electroconductivity in the laboratories with the help of conductivity meter respectively. And dissolved oxygens were measured with the help of oxygenometers. Calcium and magnesium plus ions by EDTA trimetric method, and the potassium and sodium were determined with the flame photometrimeters. So what we observed, so while collecting the sample, these are some sites photo with my guide, Dr. Anupama Pathak. And we are collected with the same pond. This is the high crystallized pond with showing the high salinity of water, the pH measures at the time of the collection of the samples, in the temperatures and the These are the ponds and these are person which help us in the near the Mulund salt. We'll move to the next. So finally, the results and discussion mostly we found the gradual increase in salinity once we move from the pond 1 to the pond 7, that is high, means the low salinity water to the high pond. It will start with the pond 1 to the pond 7 continuously. And then reduction of the dissolved oxygen from pond 1 to the pond 7 in the conditions, and the pH is observing near to the 7.6 to 7.1. These are all results are tabulated in the form, in the table format. We'll move to that table format. So these are the parameters, that is physiochemical parameters: salinity, pH, temperature, dissolved oxygen, EC, electric conductivity, TS, total solids, total dissolved solids, and all these are. So if you see the pond 1 having low salinity as compared to the pond 7 is the crystallized pond. If you move for the salinity from pond 1 to the pond 7, the fourfold increases from 9 to the 38. The salinity in percentage. pH is not much variation in the pH. The May month, the usually the temperature in the May month 37 to the 53. It's vary from pond 1 to the crystallized pond. Sorry. In the dissolved oxygens, 6.4 to the 1.6. As the salinity increases, the oxygen dissolved percentage also lower down. There is a less EC changes in all the ponds, but there is a high increase in total solid and total dissolved solid percentage. It's from almost four folds, from 98 to 433 and 80 to the 417. And also, if you see the sodium concentration from 68 to the 97 is the crystallized pond. The final, we'll move to the conclusion. Solar saltans are known as a technical diverse halophilic organism. So we are found out that it is ecosystemically important source of the Archaean halobacteria, and we are further using for our further study for my project. Thank you.
Okay. I think you have finished well within time. Is that correct?
Yes, correct.
Okay, that's good. So, Rushti or Dr. Vaishali Chaudhary, should we take questions right now, or?
Yes, sir. The session is open for question answer.
Sure. Okay. Does anybody have any questions for Mr. Bong? So, Mr. Bong, can you, if there are no other questions, can you tell us what are your expectations? Is there some sort of a hypothesis that you have, or, can you tell us a little bit more about it, because you have some time?
Yeah, thank you very much. To provide me opportunity to tell about my project. So my aim is particularly object, and for my project is that I want to produce the carotenoid producing microorganisms, archaea and bacteria from solar saltern of Mulund, because so many species found out newly in the ocean water and having a high salinity waters. So one of the source, that is natural source, we will consider as microorganisms. So once I got a good microorganism isolated from the particular site, so we'll go for the carotenoid production, and it will be very helpful for the because human disease, it will cure the human disease and anti-inflammatory reactions, because you will know nowadays we are facing so many problems due to the COVID-19. So but in that situation, with natural immunity increasing power or immunity related, we can produce from the carotenoid bacteria and archaea, it will be very good for us. So my aim is to develop the carotenoid producing archaea and bacteria from the solar salternum.
So do you know that this kind of work developing these carotenoid microbes has been done in any other specific saltons around the area, or you are trying it for the first time?
No. This is not the first time actually. I had a reference for so many sites. Even from Mulund also some are the isolates, but these are the not high producing microorganisms. So I am looking for the high production of carotenoids. Okay. So it's also in solar saltons, in the even different ocean sites have a solar salton. In Brazil also it did. So many places I find out. Okay. So it's not a new work actually, but I want to develop the high producing carotenoid bacteria and archaea.
Okay. Thank you, Mr. Bong. Is there anybody else who would like to ask anything? Okay, if not, then we will thank you, Mr. Bong, and we will move on to the next speaker. who is Ganpat Tashat Maske, who will tell us about groundwater quality from various places of Maharashtra.
Thank you, sir. Thank you, audience. Please, I'm leaving.
Can I request the speakers, if their bandwidth allows, to switch on their camera, because it is better if we can see you. Mr. Maske? You are muted.
Yes, sir. Screen is visible.
Screen is visible, but you are not visible.
Sir is asking to put your webcam on.
Yes, camera.
I will request other speakers to do the same thing because it's a little more personal that way. If you don't have a webcam, it's okay. You can start. Let's not delay too much. Yeah.
Okay, sir.
We can see you now. Let's start, please.
Screen करना है Yes, sir. I think screen is visible and I am also audible. Good morning, one and all.
Can you go into slideshow mode? Can you go into the slideshow mode?
Slideshow.
At the bottom. Right.
Yes, no?
Go to slideshow at the top. Otherwise, go to slideshow at the top.
Slideshow. Okay. Sir, am I audible?
Yes, you're audible, but you are still not in slideshow mode.
Yes, sir. Now I am in slideshow mode.
Okay. Take it. Go on.
Okay. Good morning, one and all. Myself, Mr. Ganpat Maske. I am a research student of KTHM College, Nasik, and today I am going to present a topic: assessment of groundwater quality of Jam River Basin, Sinnar and Kopargaon Tehsil, Maharashtra. I am doing my research under the guidance of Professor Dr. Mrs. Resham Balla Madam. She is from Panchoti College, Nasik. Introduction: groundwater, which is one of the important natural resource contributing significantly annual supply.
Your slides are not changing. Have you shared your full screen, or your slides are not changing?
Okay.
So you can press F5 for slideshow.
M5. M5.
F5, sir.
F5.
Have you shared your entire screen? You have to share your entire screen, otherwise it may not work.
Okay, sir, is the slide changing?
No.
No, sir.
Okay.
हाँ अभी हुआ चेंज अभी बोलिए
ठीक है ओके ठीक है okay. Today's topic of my presentation is assessment of groundwater quality of Jam River Basin, Sinnar and Kopargaon Tehsil, Maharashtra. All of us know that groundwater, which is one of the important natural resource, and in this area, which is one of the important natural resource for the agricultural practices as well as the domestic activities. But the certain factors which are continuously affecting in this area regarding the groundwater quality, that factors it including the wrong agricultural practices such as the excessive use of chemical fertilizers as well as the pesticides, as well as the by taking the same types of the crop year after year, as well as over irrigation, as well as the dumping of the waste material directly onto the land. Such activities continuously affecting the groundwater quality, as well as there is found continuous soil and water interaction, and due to the dissolution of certain minerals, as well as the by the various anthropogenic activities which are affect the groundwater quality. Then my study area is the Jam River Basin. Jam River, which is a tributary of Godavari River, it originates at Masha Mountain of Sinnar Tehsil, Nasik district, and confluence to Godavari at Kopargaon of Ahmednagar district. It flows 52.2 kilometers from the eastern part of Sinnar Tehsil, and basin extends to southwest part of Kopargaon Tehsil of Ahmednagar district. this area includes the total 94 villages are there, and area size of this area is 636 square kilometer. Then for the purpose of sampling, I have used the grid method. Total this blue color boundary shows the boundary of Jam River Basin. This is the total 636 square kilometer area. It includes some part of Sinnar Tahsil, some part of Kopargaon Tahsil, as well as some part of the Sangamner Tahsil. Then total 94 villages are there, and I have made certain grids. Total 60 grids are there. Size of each grid is a 3.25 by 3.25 kilometer. Therefore, area of each grid 2 square kilometer. And I have used this method, a grid method, for the purpose of sampling. Then, objective of my study to conduct survey of study area and mapping of sampling sites using GIS to monitor groundwater quality in post-monsoon season 2020 and to suggest control measures for the improvement of groundwater quality and quantity in the study area. Then methodology, including the three important points, including number one, selection of sampling site by using the GIS. I have made the whatever the grid method. Then for its whatever the physicochemical analysis. Then collection of the samples. For the purpose of collection of samples, use the plastic container having capacity 1 liter, and use the plastic containers having capacity 1 liter. Then at the time of sampling, some of the parameter I have tested in the at the field, it including the pH, AC, temperature, as well as the well depth, as well as the static water level, lithology exposed, operating condition of the well, as well as the GPS location of that area, as well as the altitude range of that area. Then methods for the physicochemical analysis. These are the photographs of sampling of the water sample. I have collected the water samples from the wells as well as the bore wells, and from the wells, after pumping pumping it for 30 minutes, and from the bore wells collected the sample after pumping it for a few minutes. Then physicochemical parameters and the methodologies. These are the certain physicochemical parameters which I have analyzed during the 2020, and the methods used for the analysis of that parameters. For some of the parameters, digital thermal, some of the parameters, whatever the electrometric method. Then for temperature, digital thermometer. Then for some of the parameters, titrometric method I have used. Then for the nitrate, nitrogen, nitrate. Use the method spectrophotometric method, nitrogen and ammonia Jaldar's method, orthophosphate, total phosphate and sulfate, use the method spectrophotometric method. Then for the sodium and potassium, use the flame photometer method. See, for the different parameters, use these methods, and my focus or important focus is regarding the result. These are the results. I have analyzed the water sample post monsoon during the post monsoon season. Different first column shows the different parameters. Then this one column shows the range of results. Then in this column here is shown the high range sample numbers. Here is mentioned only the sample numbers. Then desirable permissible limit for the drinking of BIS 2012, and irrigation permissible limit for the irrigation water permissible limit of Food and Agricultural Organization 1985. See, pH for the groundwater analysis during the post-monsoon season 2020, pH found in the range of 7.2 to 8.7. Means all the parameters excluding only one, that is the all the samples excluding only one sample number, that is the 34, which is above the permissible limit for the drinking as well as the irrigation purpose, but remaining all parameters are within range. Then EC found in the range of 100 to 937. All the samples sampling stations show the EC range within permissible limit. Then calcium 18.04 to 113.34. Calcium also shows within permissible limit at all sampling stations for the drinking purpose as well as the irrigation purpose. Then magnesium, its range found 14.35 to 115.86. Sample number 34, 5 and 12 shows the high level. Then out of that, sample number 34, which is above the 34, 5 and 12 12, these three shows the above the permissible limit for the sodium 27 to 347.4. Again, the same sample number shows they are at the higher level for the drinking, but these are also within the permissible limit for the irrigation. Then potassium 0.5 to 11.4, same sample numbers which are above the permissible limit for the irrigation purpose. Chloride, all the sampling stations are within limit. Then sulfate, also all the sampling stations are within the limit. Then nitrate,
Sir, you have 2
I have
minutes
found
left.
that. Okay, madam.
Sir, you have 2 minutes
See,
left to end up.
yes, madam. See, my study area is broadly divided into the major under the two headings, that is the irrigated and the non-irrigated. See, in the irrigated area, which is near the Kopargaon, that is the irrigated area, it shows the high amount of the nitrate level for most of the sampling stations. Then fluoride within limit, total hardness for these three parameters that are above the limit for the drinking, but are within the permissible limit for the irrigation. Then TDS within limit, and carbonates high level for the irrigation standards. See, these all the major ions and TDS are expressed in milligram per liter, while pH on scale and EC in microsiemens per centimeter. Then in the irrigated area, they are found such kinds of the problems. That is, after the irrigation, they are found the whitish layer on the soil surface, that is the layer of the salt. Then conclusion of my research is the groundwater quality is significantly affected and found vulnerable due to intensive agricultural and anthropogenic inputs in irrigated area. Outcomes, including the analysis of river basin groundwater samples, will serve as a baseline data to help in formulation and management of action plan for the long-term conservation of natural resources, and study will immensely help for the farmers to select the cropping pattern in the study area. An analysis of groundwater and soil quality will help in the improvement of agricultural production in the study area. This is my research work. Thank you.
The session is open for question answers. Anybody want to ask any question to Maske sir?
Okay, great. So, do people have anything to ask Mr. Maske? Anybody has any questions for Mr. Maske? Okay. If not, do you plan to repeat the same studies, let's say in the summer or in some other at some other time of the year, Mr. Maske?
Yes, sir. Actually, in my research work, 2 seasons I have selected, that is the pre-monsoon and the post-monsoon. But these are the readings of only the post-monsoon season, and after a few months, I am going to analyze the all the parameters for the pre-monsoon season also.
Okay, what changes do you expect? Do you have any expectations? What is normally known?
Sir, I am expecting that there is increase. There should be increase in the TDS, EC, as well as the hardness, as well as the nitrate content, cationic components during the pre-monsoon season.
Okay. Okay. Good luck with your studies, and I hope you will be able to complete them in different seasons as well and arrive at a conclusion. Thank you, Mr.
Yes, sir. Thank you, sir.
Okay. We next have Aisha Amin Jumani. He's going to tell us about the effects of certain disinfectants on bacteria.
Yes, sir.
Are you online?
Yes, sir.
Yeah. Okay. Share your screen, please.
Okay. Is the screen visible?
No, Aisha.
Okay, ma'am. Is it now visible?
Yes.
Okay.
Aisha, put your webcam on.
Yeah, ma'am.
And also go in the presentation.
Just a minute. There's a glitch. I don't know what's happening. So can you give me a minute, please?
Sure.
हाइड करो क्लियर से पूरा करिए फाइन yeah okay.
Is it now visible?
Yeah, please go on. Yeah.
For webcam.
Just switch on your camera at the bottom.
Yeah, sir.
Yeah, okay, please start.
Okay. A very good afternoon to everybody present here. This is Aisha Jumani, and today I'm going to speak on the topic study of efficacy of disinfectants on potent bacteriological species. Now, we know as microorganisms are present everywhere and they are not visible through our naked eyes, but the microorganisms present have different functions. Some are beneficial, some may cause harm to us. The human body also has a wide variety of microorganisms or bacteriological species present inside us, but some of the microorganisms are parasitic in nature. That is, they depend on the human lives, and they harm to the human and they live their lifespans. So in order to cut off that microorganisms and to maintain the sanity of ourselves as well as the environment, it is very necessary to take actions and precautions for a better lifestyle. So my project depends on my project actually focuses on the nosocomial infections, that is the infections that happen in hospitals. So hospitals usually get in hospitals, we can see there are a lot of patients admitted and the immunity is already low into them, and so in that condition the bacteriological species they get great opportunity to act onto them and to infect that person. So to reduce that percentage, we use sterilants, disinfectants to decrease the amount of bacteria present there. So there are two types of solutions which are used for cleanliness purposes. But the first one is disinfectants. The name itself suggests that these solutions are used to destroy microorganisms, but they cannot eliminate them. Some amount is present, but and the other is sterilants. The sterilants are used to eliminate the spores, the bacteria, viruses, fungi, everything. In comparison to disinfectants and sterilants are widely effective. In my project, I have used disinfectants such as Dettol. Then I have a photo. It's Dettol, Lysol, and sanitizer. Here I'm going to compare the three of them on the bacteriological species like Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa. So these solutions are not directly used. Working solutions of the disinfectants are prepared. So how are the working solutions prepared? We created some percentage like 25%, 50%, and 75%, to which the distilled water is added and they are tested. Firstly, we have to obtain the pure cultures of the microorganisms used. The pure cultures are prepared in nutrient agar plate, and then pour plate method is performed. Pour plate method is basically where we add cultures to the hot nutrient agar, and the cultures are added, and it is plated into a sterile plate, and from using a cork borer, using a sterile cork borer, the wells are created, and the diluted or the working solutions of 25%, 50%, and 75% working solutions are added into the well according to the suitability of the microorganisms to be grown. They are incubated at room temperatures or in hot air oven, and we wait for 24 hours till we get the growth. In the well where we add the disinfectant solutions, it creates zone of inhibition. Zone of inhibition is what we can see in this picture. The first one is the plates of Escherichia coli. We have 3 different plates. The first one is of Dettol, the second one is of Lysol, and third one is of sanitizer. We can clearly see the circular zones of different concentrations. As the concentration increases, the zone of inhibition also increases. Same as of Staphylococcus aureus and same and of Pseudomonas aeruginosa. Amongst the 3, the Pseudomonas aeruginosa shows the least effect of the disinfectants, and the sanitizer and the working solution of this sanitizer shows no effect. Here is the table, and there are some numbers of diameters of zone of inhibition. As the diameter of the zone of inhibition increases, it shows that the which disinfectant is more effective. We have in this research, we have used 2 types of methods to conclude our result. The first, which I explained earlier, was the agar cup method. The second comes the use dilution method. The use dilution method is basically used on inanimate objects, where the steel cylinders are used. The steel cylinder is first sterilized. It is put into the solution of the culture, that is Escherichia coli and other 2. Then it is dried off, and it is put into the working solutions of disinfectants, which is taken out from the test tubes, and it is put into the sterile nutrient agar and incubated at the suitable temperatures. When the 24 hours of time is achieved, we see the culture's consistency. As you can see in the picture, the last one is the positive control, through which we'll conclude the other 4 test tubes and we'll conclude the result. The first and the second test tubes shows the turbidity, which means the growth is achieved. And now the use dilution method is not quantitative, it is qualitative. That which means here we are not counting the colonies or not counting the number of microorganisms which grows, but here we are seeing whether the growth has taken place or it is not taken place. The second, the third and the fourth tubes does not show the growth, which means the disinfectant was able to cut out the growth of the microorganisms, whereas in the first and the second, the growth is achieved. That means the disinfectant was not that effective that it could eliminate the growth of microorganisms. Here is the chart which shows Dettol shows the highest efficiency towards killing of the microorganisms, whereas sanitizer shows the least. My conclusion of the project is sanitizers are least effective in the COVID times. Before COVID, sanitizers were not in huge use, but according to my preference, since COVID times, the use of sanitizers is increased heavily without knowing its history, without knowing in detail that it is least effective. And apart from the research work, one more topic which I want to introduce here is there were many incidents happen in the COVID times because of excess use of sanitizers. There's burning of kitchens, burning of vehicles, excess use of sanitizers on kids, which caused them lungs diseases. So I hope my project creates a light on further studies in detail about the sanitizers before using it excessively.
Ma'am, we have 2 minutes left.
I'm done, ma'am. Thank you.
So the session is open for question answers. Anybody wants to ask question to Aisha?
Yes. May I?
Yeah, please go ahead.
Okay, Aisha, since you have analyzed or compared three things, one is Dettol, one is Lysol, and the other is sanitizer, but all of have multiple compositions. And so from your research, would you say that the use of sanitizer is useless? Means we are in the wrong direction.
No, ma'am, I did not intend to say that. I'm just saying I compared three of them. Giving the recent example, in COVID times, we are using sanitizers everywhere, like literally everywhere. So, is it advisable? Is it necessary? Is it required to use in such excess manner? So, for the study is required. This is what I meant to say.
Okay, so sanitizer is basically alcohol. So you could have taken one more control as only alcohol to see whether it is the effect of the alcohol or any other thing that has been added to it.
Ma'am, basically when we do...
Similarly, Lysol also has many components in it. So whether it is because of the main component or because of something which has been added,
yeah,
that is not clear. That is not clear.
Okay, shall I speak, ma'am?
Yes.
Okay. So, as you said, I could have taken one alcohol control. So, in bacteriological practicals, we know when we use 100% alcohol, it is ineffective towards bacteriological species. So, either we have to use 95% or 75%. That was the reason I used sanitizer, because it has some 90% of alcohol concentration in it.
Yeah, that's fine. But again, sanitizer does have some addition. So it would have been preferred if you had used maybe say 95% alcohol also. There should have been some control. I feel so.
Okay, ma'am.
Thank you. Thank you.
Okay, are there any other questions for Aisha? Okay, if not, thank you very much, Aisha. We will move on to the next speaker, who is Piyush. And Piyush will tell us about how to use snake venom against the coronavirus.
Thank you, sir.
Good afternoon, everyone. Is my voice audible?
Yes.
Okay. Okay. Shall I start, ma'am?
Yeah. Start, please.
Can you begin the slideshow, ma'am?
Yes.
Okay. Good afternoon, everyone. Respected judges, senior researchers, principal madam, and all the dignitaries present over here. My name is Piyush Bharadwaj, and I am a student of MSc Biotechnology from Rajiv Gandhi Institute of IT and Biotechnology, Bharati Vidyapit, Pune. Along with that, I am a wildlife rescuer. I have been rescuing snakes and different reptiles since 5 years. So I knew the importance of snake venom, what are its components, and how it can work. So this was a review I made in my TY BSc when the COVID pandemic hit India. So the title of my today's presentation is Mechanism of Biomolecules Extracted from Snake Venom as Antivirals to Inactivate Novel Coronavirus. The abstract. Snake venoms are used as defense mechanism or to immobilize and digest prey. Ma'am, next slide, please. Snake venoms are used as defense mechanism or to immobilize and digest prey. Venoms are complex mixture of enzymatic and non-enzymatic components with specific path physiological functions. Peptide toxins isolated from snake venom mainly target ion channels, membrane receptors, and components of the hemostatic system with high selectivity and affinity. Consequently, snake venoms can be considered as mini drug libraries in which each drug is pharmacologically active. However, this time of crisis is generating an urgent need to develop therapeutic applications to tackle the current pandemic. The considerations presented in this document reveal the use of biologically active components isolated from snake venom as potential source in drug discovery to inactivate the novel coronavirus. Ma'am, next slide, please. Snake venom is composed of a mixture of proteins, peptides, 90 to 95% free amino acids, nucleotides, lipids, carbohydrates, and metallic elements coupled to proteins. Some studies have reported the antiviral activity of snake venoms and their components against measles virus, Sendai virus, dengue virus, yellow fever virus, and HIV. Thus, snake venom are sources of promising candidates for new antiviral drugs. The present research is based on an up-to-date survey on the pharmacology of snake venom bioactive components and evaluates their therapeutic perspectives.
किसी और से बात करके मैं थोड़ी देर बाद फोन करूं. अच्छा जोएल, हाँ तो tell me what happened. So they have done an RT-PCR and you are positive, right? And now you have taken you to a specific hostel where they have kept you under quarantine, right? ठीक है. और उन्होंने अभी तुमसे ये पूछा है तुम किसके साथ कांटेक्ट में आए थे वगैरह, ये सब का क्या प्रोटोकॉल हुआ है? हाँ ओके ठीक है. तो तुम्हारा फ्लोर तो शायद ऑलरेडी सील्ड होगा, राइट? क्योंकि
Just wait for 2 minutes. He's on another call. Okay?
Right?
Yes, ma'am.
Don't get worried. Huh? Piyush,
Yes, ma'am. Yes, ma'am.
don't get tensed. He's on another
No,
call.
no.
He'll join us. Okay? Don't go. Just relax. Okay, Piyush, you can continue with your presentation.
Okay, ma'am. I'll begin with this slide again. Snake venom is composed of a mixture of proteins, peptides, 90 to 95%, free amino acids, nucleotides, lipids, carbohydrates, and metallic elements coupled to proteins. Some studies have reported the antiviral activity of snake venom and their components against measles virus, Sendai virus, dengue virus, yellow fever virus, and HIV. Thus, snake venom are a source of promising candidates for new antiviral drugs. The present research is based on an up-to-date survey on the pharmacology of snake venom bioactive components and evaluates their therapeutic perspectives against a wide range of pathophysiological conditions. L-amino acid oxidases have become an interesting source of biomedical research because of their anticoagulant, antimicrobial, platelet aggregation inducing and inhibiting, apoptotic inducing, and anti-cancer activities. Phospholipases A2, PLA2's enzymes, which play essential roles in various biological activities, are common to many snake venoms. Phospholipases are a class of enzymes that have the ability to hydrolyze specific substrates like glycerophospholipids. Ma'am, next slide, please. LAO, that is L-amino acid oxidases, isolated from Bothrops jaraca venom, reduce the viral load in cells infected with dengue virus exposed to the toxin. These flavoenzymes also produce hydrogen peroxide H2O2 as a free radical, which appears to enhance their antiviral activity. Phospholipases A2 interact with the host cells and prevent the intracellular release of virus capsid protein, suggesting that they block viral entry into the cells before virin uncoating. Antiviral activity involves the cleavage of the glycerophospholipid virus envelope and protein destabilization, on which partially exposes the genomic RNA and culminates with viral inactivation, making it unable to access the cell receptor. Metalloprotease inhibitors, which could prevent the production of new HIV particles by inhibiting the viral proteases. In addition, immunokine, an oxidized derivative of alpha toxin extracted from Naja siemensis venom, has been shown to inhibit infection of lymphocytes by HIV through chemokine receptors. Mucoporin M1, another example of the antiviral effect of biomolecule, presented antiviral activity against measles, SARS-CoV, and influenza H5N1 viruses, possibly through a direct interaction with virus envelope. Ma'am, next slide. The methodology. The protocol is mainly based upon 7 steps. First is source of venom extraction. In India, there are various diversity of venomous snakes found. Then the second step is isolation of bioactive components from venom, purification and dilution of the components, cultivation of viruses, testing procedure, method for formulation, and mode of therapy. Ma'am, next slide, please. The result and conclusion according to my observation and review, we can also the LAOs, that is the amino acid oxidases, inhibited HIV-1 infection and replication in a dose-dependent manner. In the same way, LAOs have the potential to inhibit the replication of coronaviruses. Also, LAOs produce hydrogen peroxide as a free radical, which enhances the antiviral activity. The phospholipases A2, PLA2 have the potential to interact with the host cells, and they can prevent the intracellular release of coronavirus capsid protein, suggesting that they block viral entry into the cells before uncoating of coronavirus. The mechanism proposed for PLA2 CDT antiviral activity may cleave the glycerophospholipid of coronavirus envelope and may destabilize the spike protein on the viral surface, which partially exposes the genomic RNA and culminates with viral inactivation, making it unable to access the cell receptor. The metalloprotease have the potential that it can prevent the production of new coronavirus particles by inhibiting the viral proteases. We can also use laboratory-based solid phase peptide synthesis to synthesize bioactive molecules in large quantity. This was a review I made in the month of March-April after my TYBSc. After that, I presented this paper in an ideathon by ICMR and I was selected too, but I couldn't do any laboratory work due to the circumstances. But after that, ma'am, next slide. There were further advances made in this field after a year almost. There were articles like Brazilian vipers venom can stop COVID-19 from multiplying. Scientists discover snake venom that kills COVID-19 with no harm to humans. Bioactive molecules derived. These studies were further made and it was shown to be successful. Ma'am, next slide. This is a reference for my review. Next slide, ma'am. Thank you.
Okay, thank you, Piyush. Thank you, anyone has got questions for Piyush?
Any questions for Piyush? So, Piyush, let me ask you, can you hear me?
Yes, sir.
Yeah, so what you're proposing is very interesting, but practically snake venom is not easy to obtain in large quantities, right? Compared to that, you know, using recombinant technologies, you can make vaccines, antibodies in much larger quantities. So do you think what you are proposing will, you know, it's interesting, but practically how feasible is it?
Sir, actually, this was just a review, like idea, like it can work. Actually, if we can try, it is practically impossible, but new approaches and technologies of venomics to meet challenge, it can be successful if we try some experiments possible, then we can check, like we can try it and further we can see the results if it's possible or not.
Okay, thanks. And good luck with your work that you are doing and you're rescuing snakes. That is very good to know that. Good luck with that. Okay.
Thank you, sir.
Thank you. Okay, from Piyush, we will move to Rushti Ritesh Patel, who will tell us about characterization of eucalyptus oil extracts.
Good afternoon, sir.
Okay, Rushti, can you share your screen?
Yes, sir. Is it visible? Hello,
yeah,
sir.
please start. Yeah. And are you going to time yourself?
I have given it to someone else.
Okay.
Is my slide visible? Shall I start, sir?
Yeah, it's visible. Please start.
Myself, Rushti Patel, and I will be presenting on extraction and characterization of essential oils from Eucalyptus globulus leaves by 3 different methods, that is hydrodistillation, Soxhlet apparatus, and steam distillation. So in this growing pandemic, we all have known about the importance of immunity, how important it is to have being immune properly. So our essential oils are the ones who are definitely going to support our immune system. Talking specially about Eucalyptus globulus leaves, it is comprising more than 750 species and subspecies which are indigenous to Australia. Now, particularly, the leaves are those part of this particular tree which are having the active biological secondary metabolites present in them. So there are almost 204 non-volatile constituents present in the leaves of these species. So I've listed these are some of the chemical constituents which are present in eucalyptus oil and their uses. So we can see they have been used maximum in perfumery flavoring, as a raw material for medicinal purposes. They have been used as an additive for industrial perfume solvent, as a fixative, as a bactericide. 1,8-cineol, that is listed second in the list, is the maximum component of our eucalyptus oil, which has been widely used as medicinal and cosmetic purposes. We all know eucalyptus as a Nilgiri oil also. So these are the specification of that oil. According to specific gravity, it is 0.905 to 0.925. Its appearance will be colorless to pale yellow liquid. The purity, that is almost 1.18 cineol is contained, almost 80%. The optical rotation goes to -5 degree to +10 degree, and the refractive index 1.458 to 1.470. So what are exactly the essential oils? Essential oils are those volatile aromatic compounds which are present in plants, and they are responsible for giving a particular aroma to your plant. So they are actually the heart of the plant which are giving them wonderful scents. So we can use them for body oils, compresses, cosmetic lotions, hair rinses, perfumes, etc. So right now there are several methods of extracting essential oils, but I've worked on only three, that is Soxhlet, hydro, and steam. So our first method will be the steam distillation. As the name goes, we are using steam for extraction of our essential oil. The steam will be generated in the steam boiler, which has been controlled. The temperature of the steam boiler is being controlled. The steam is passed on to the steam jacket that is present, inside which our plant material will be placed. The care is to be taken that plant material will be placed on a perforated grid so that it does not come in direct contact, or it will not get charred due to the heating temperature of your jacket. The volatile components are collected, they will be evaporated along with the vapors, and they are collected into the condenser. So after condensation, along with the condensate water, the oil will be given out into the oil separator. So oil being lighter than water, it will be collected in the upper part, and the condensate water can be removed from the downward part. So the procedure goes that we have collected the leaves, we have cut it into 2 by 2 centimeter, we have weighed 100 gram. The care is again to be taken that within half a day after collection, you are going to carry out your processes. The distillation was carried on for 5 to 6 hours, and then it was ceased. And then we moved for calculating the volume of essential oils that was determined. So as I earlier told, the precaution is to be taken that the temperature of the extraction chamber should be controlled. It should not be much high, otherwise it will result into harsh aroma rather than the floral aroma. And then it will affect your therapeutic effects of our oil. The next method is the hydrodistillation, almost going parallel to steam. But here we are soaking our plant material along with water, so it is physically mixed into the boiling water. The water is boiled either with the help of firewood, which is a traditional method. But if we are carrying it to laboratory, we can use our Bunsen burner also. So plant material will be again boiled along with water. The vapors that are evolved, the volatile components that are evolved will be collected into the condenser. And again, they will be sent to the oil separator for better separation. And again, we will carry out the calculations, that is volume of the essential oil that was determined. The process will take almost 4 to 5 hours. So we will cease the process after 4 to 5 hours. Again, a precaution is to be taken that we are putting some extra water or enough of water so that the duration of distillation at least lasts for a particular time. Otherwise, there are chances that our plant material will get overheated or charred. The last method that was studied was the Soxhlet apparatus. So here we are doing a kind of a solvent extraction where we are using hexane as the solvent. So as we can see, we have got the apparatus distributed into three parts. We have got a proper Soxhlet apparatus, we have got a heating mantle at below, and the upper part is our side siphon arm or a condenser tube. So in this case, these are our these are our materials. Now in this process, rather than cutting, we will use the grinded eucalyptus leaves. I have weighed 100 grams of the eucalyptus leaves. We have prepared a thimble using a filter paper. So we will insert our grinded eucalyptus leaves inside a thimble, which is made up of a filter paper, and then place it onto a Soxhlet extractor. So the second part is our Soxhlet extractor. Below that, we have a hexane solvent present in the heating mantle. So using heating mantle, we will allow the hexane to boil. So as the hexane boils and vapors come out, it will move through the Soxhlet extractor. All the volatile components along with the oil will move along with the hexane and they will be condensed. So we can regain that particular hexane solvent again back into the heating thimble. What actually is happening is that hexane is refluxed. So the solvent vapor travels up a distillation arm and flooded back into the chamber. So we can again reuse that hexane solvent again and again. That is, it is not necessary that you have to put it some other warming solvent. So this is one of the solvent again and again. So the thimble was changed only after making it run for many continuous cycles. So we will continue it for a few cycles. I've continued for 15 cycles. So after 15 cycles, the desired compound is concentrated in the distillation flask, and that can be removed and can be considered as a one batch. So the volume that was obtained was measured. We carried out the necessary mass balance, that initially how much hexane was used and how much hexane been lost in the process, that was calculated. So that was the mass balance. The yield, the moisture content was also calculated, and the percent oil recovery was also calculated. For purity, it was outsourced using gas chromatography. So this was the result that we got while comparing the three distillation processes. So we saw, compared to hydro and Soxhlet, the best purity was given by steam distillation. So these are my results that I've kept it very common that starting raw material should be 100 grams for all. The weight of oil received for hydro distillation was 4.2. So the percentage is 4.2%. Soxhlet gave me only 4.6 gram, whereas steam distillation gave the highest, that was 6.1 gram. So the characterization was done. The specific gravity was measured using specific gravity bottle. Optical rotation was measured. So using the polarimeter, the refractive index was measured using refractometer. So compared to the literature, almost all the specifications, all the characterizations were matching. So eucalyptus oil can be used as a co-solvent, antiseptic, expectorant, and diuretics also. It has been used for anti-inflammatory processes. So my conclusion is steam distillation is a special type of distillation that has been used by wide industries, and it has been the choice for carrying out extraction of essential oils. So it is specially designed for temperature sensitive materials like oils, which are insoluble in water and may decompose at other boiling points. So the experiment has been carried out for extraction of eucalyptus oil, which have been used as a perfume, chemical raw material, and the studies are now been done for considering it as a renewable sources of energy. So my future work includes that the eucalyptus oil contains more than 65% of 1,8-cineol, and hence the incorporation even if small amount of eucalyptus oil as a co-solvent in aqueous ethanol and petrol mixture will improve the water tolerance of the system. So the work can be extended if we continue to study the ternary phase equilibrium of water ethanol and 1,8-cineol or eucalyptus oil. So that can give us a better option for saving our environment from pollution. So these are my references. Thank you, sir.
Thank you, Rushdi. People have any comments, any questions? So you mentioned about the oil, the refractive index and the other properties of the oil. Did you check that for the other methods of distillation, and are you getting equal purity, etc. of oil.
Rup sir, please unmute yourself.
I am unmuted. Can you hear me?
Yes, sir. Yes, sir. I can hear you.
Yeah. Did you hear my question, or should I repeat?
Yes, sir. Yes, sir. I heard your question.
Yeah.
I checked it for all the three methods, sir.
Okay.
So the best results were given out for steam distillation.
No, the results in what sense? Results in the sense
Results,
of, yeah.
the first, that in the sense of characterization, that the specifications which were mentioned in the literature, and the most important, the results of the GC that were outsourced for the purity.
Okay, so you got better yield and you also got equally good or better purity. That's what you're saying.
Sir. Yes, sir.
Okay.
With the help of steam distillation.
Okay, thank you. Thank you, Roshni. We move to Harshita Pandey, who it says it's a poster, so I don't know what that means.
She will be giving a poster presentation. She will be explaining her poster, sir.
Okay. So do we have a break, or how long do we go on?
Do you want break? Do you need break?
No, it's okay. We can continue. So the last one is, about 6 or 7 more talks. Okay, let us go on.
Thank you, sir.
So my screen is visible?
Yeah.
Yes, sir.
And audible too?
Yes.
Thank you. Good afternoon, everyone. I am Harshita Pandey. Today I am here to present my poster on cytogenetic finding of pediatric acute lymphoblastic leukemia patients. Leukemia is a cancer of hematopoietic system that affects white blood cells. Normally, white blood cells grows, divide, and reproduce in an orderly and controlled manner. When uncontrolled cell division occurs, it leads to leukemia. Leukemia leads to the suppression of normal hematopoiesis. Leukemia can be classified as acute or chronic and myeloid and lymphoid. That is acute lymphoblastic leukemia and acute myeloid leukemia, chronic lymphoid leukemia and chronic myeloid leukemia. The term leukemia was given by Dr. Rudolf Virchow in 1847. ALL is a cancer of blood cells which affects white blood cells called lymphocytes. The reason behind calling it as acute disease is that it spreads very quickly, and if not treated, it becomes fatal within few months. Genetic factors have a significant role on the development of childhood ALL. Recent advances in cytogenetic have led to better understanding of important genes that are involved in leukemogenesis. Conventional cytogenetic analysis has been the standard method of identifying genetic aberration, including changes in chromosomal structure and number. Genetic abnormalities play a major role in prognosis and treatment outcome of ALL patients. Routine chromosomal analysis or karyotype refers to the study of chromosome at metaphase stage. The aim of my study is to identify different chromosomal abnormalities in 200 pediatric ALL patients by using karyotyping. Methodology: we have taken 200 peripheral blood and bone marrow samples of diagnosed pediatric ALL patients. For cytogenetic studies, samples were cultured using standard culture techniques and cells were arrested at metaphase by using colcimate and then processed further. Cells are then dropped onto the pre-label slides and then gene subbinding was performed. The result of my study was out of 200 cases, 127 patients were male and 73 were female. We have obtained 188 successful cases out of 200 cases. 37 cases exhibited abnormalities and 151 cases have shown normal karyotypes. Both numerical and structural abbreviations were observed and 5 cases have shown the complex karyotypes. Numerical abnormalities were found in 24 cases, that is 12.76%, and structural was obtained in 8 cases, that is 4.25%. Out of 8 structural abnormalities, we have found translocation 922, which was observed in 6 patients, and translocation 822, which I was found in 8 patients, and translocation 1221 was also found in 1 patient. The conclusion of my study was we observed different chromosomal aberrations in different age of children and also frequency of various chromosomal structure and numerical abnormalities in ALL. Thank you and have a nice day.
Okay, is that all? Okay.
Yes, sir.
Thank you. Thank you, Harshita. You went quite fast. So, are there any questions from anybody? All right. If not, we will thank you again, Harshita, and we will move to Dr. Sharon D'Souza and Dr. Sagarika Gamble, who will tell us about molecular profiling to determine diversity among ethnobotanical plants from the Western Ghats.
Sorry for my background. I'll just share my screen. I'll try to do it through slideshow, sir, but I'm not sure if that is going to work. Can you
Yeah,
see
we
my?
can see your slides. Please go on.
Can you see if it is moving, sir? Is it showing?
No, it's not moving.
Yes, sir. So that's the problem actually. I will try to do this full screen. Is this fine?
Yeah, now try to move this, change the screen slide.
Yes, sir.
Yeah,
Yes.
it's changing now. Yeah.
Okay, thank you, sir. Thank you so much. Uh, uh, Good afternoon, everyone. I will be presenting a part of our PhD project that we had done. That is molecular profiling studies as to determine genetic diversity amongst ethnobotanical plants from Western Ghats and their urban counterparts from urban counterparts from Rubiaceae family. This was done under the guidance of Dr. Sagarika Damle. Just a brief about our work. What we had done was we wanted to study the ethnobotanical plants along the Western Ghats, and ethnobotany is the study of those plants that are used by the tribal people. And for that matter, we had targeted 3 locations along the Western Ghats. One is Nasik from Maharashtra, wherein we targeted Surgana. The places where you see yellow color flag is where we have sampled the plants from. So there we contacted the Bhoi tribe, and in Goa, Satari in North Goa, there we contacted a forest dweller, and in Karnataka, we targeted Karwar, which is in Uttar Kannada, and there also we contacted a forest dweller who is a Vaidya. So in our study, we wanted to establish the importance of studying these plants. For that matter, what we targeted was we did a phytochemical studies, bioactivity studies, and genetic diversity studies, of which today I'll be presenting to you the genetic diversity studies. So just a gist of how we went about was we did a survey first in which we recorded 82 plants and their ethnobotanical uses, of which we authenticated 17 plants, and of which we went on to select the 3 plants that we were going to study for our this PhD project. So what was the rationale for our study of genetic diversity is that when we talk about ethnomedicinal plant, we talk about it from a particular region, the original site, which becomes the tribal area, wherever they have settled down. Now, to study that, we need not just once, but we need multiple number of samples from the same site in order to do all the phytochemical, bioactivity, and all that studies. So what happens is there is continuous foraging of those plants, because of which there's depletion that happens of those plants in the ethnobotanical site. So then can we do an alternative wherein we find a different location where it is present and is present in abundance also. But when we talk about different locations, then what comes into picture is geographical locations and their habitats, because they differ. So then will they affect the phytochemical, bioactive, and genetic makeup of these plants? So we wanted to do that studying of these factors to determine whether such an effect exists. So then once we are doing that, what we could also do is that we strategize this for both research and commercialization purposes to avoid disturbing of the hotspots and select an alternative location. So our hypothesis was plants growing in rural habitats or sacred groves should have greater number and higher concentration of bioactive compounds. But if the composition did not differ much, then can we consider plants from either of the geographical location as source of pharmaceutically important bioactive molecules? So our aim was to do this analysis using gene homologue markers and fingerprinting of the selected plants to assess the genetic diversity. So So, genomic DNA comprises of both the types of genes, ones which are responsible for phenotypically evident characteristics and those which are not. So when we talk about taxonomy, then we look at the phenotypical characteristics, and that's how the authentication goes about in the herbarium. But when we talk about genomic DNA, they also comprise of those genes which are not responsible for the phenotypical characteristics. So amongst them, the ones which do not code for phenotypically evident characteristics, there are few, but they may be very important for the normal functioning of the plants. So the markers that we are choosing, and of course it is generally used for barcoding, that is MATK and RBCL. So using such markers, one can assess the diversity that exists at the foundation level and not merely the phenotypic characters. So going on further, we selected these 3 plants: Myna spinosa from Nasik, Catenarigum spinosa from Goa, and Ixora from Karwar. They all have ethnobotanical significance. Myna is used to treat inflammation. Catenarigum is used to treat jaundice. Also, it's like a fodder for catching fish. And Ixora, the roots were used for treating herpes. And what we also did was we selected in order to study the geographical, the effect of geographical location, we happened to also find these plants in and around Mumbai. So in Mumbai was considered as the urban counterpart. So all in all, we had a pair of these plants, that is one which is collected from the original site and one which is collected from the urban counterpart. So we devised 2 studies. One is 2 methodologies. One is we did RAPD analysis, and the other was barcoding. So for RAPD, we targeted 5 primers, and we used CTAB, Doyle and Doyle CTAB method for extraction. And after doing so this was done by PCR, and this was the temperature conditions. I'll not go too much into this, but once we obtained the PCR results, what we did was we did a band scoring and binary data generation using PyElf, and then we created a dendrogram using PopGen program using the the binary data that was generated. For DNA barcoding, the extraction part was the same, but where we differed was the PCR, wherein we used the gene-specific primers of MATK and RBCL. And then we sent the amplicons for sequencing. And once the sequence was obtained, we annotated it and submitted to NCBI, which is now all available, 6 of MATK and 6 of RBCL.
Ma'am, 2 minutes left.
Okay, so we did a phylogenetic analysis of DNA barcoding data using Mega6. So these were our results. When we used PopGen software analysis, they were divided into different populations, the samples, and when this is the dendrogram that we obtained. In short, we should have got 3 clades, because it was Ixora from Mumbai and Karwar, Catenarigum from Mumbai and Goa, Myna spinosa from Nasik and Mumbai. But what you see are just 2 clades. And what has also happened is that Catenarigum, if you see the population 3, has deviated from the ancestral node itself. So another confirmation here is when we treated it as groups, also Catenarigum spinosa from Mumbai and Goa showed the highest distance. Further DNA barcoding studies, when we did the phylogenetic tree using Mega6 analysis, what difference we saw was Myna spinosa from Mumbai and Nasik deviated at the ancestral node and did not form a clade. And Minka major we used as a outlier. And the same was confirmed by RBCL sequences. Because these both are protein coding sequences, we went ahead and took the liberty to do a protein structure analysis. First, we built the protein structure in silico with the help of a template in Swiss model XPC of MATK, and then we aligned it with each other. That is Myna spinosa MATK sequence of Nasik with Mumbai, and so on. So when we did that alignment, we saw a lot of gaps in between, and the homology was it varied between 60 to 70% for MATK all in all for these 3 plants. And RBCL sequences showed complete homology that showed that it is actually a conserved gene, which is absolutely required by the plant, and mutations are like almost nil. So So this is the
Ma'am, your time is up.
okay.
Please
This is
conclude
the
your time.
dendrogram, and I like to just conclude saying that as I had said that only genomic diversity has been studied here. We have done many other studies, and to correlate with what we have obtained, we had done a soil analysis studies wherein we had also done like micro and macronutrient analysis from the soil that were collected from the respective spots where the samples were present. And these micronutrients like iron, molybdenum, copper were all found to be exceedingly present. And because we know that these enzymes require, that is the secondary metabolite enzymes require cofactors to function, this could be one of the correlation that we could present. And yes, my time is up and I will try to conclude. These were our project
Please try to
contributions.
conclude fast.
Yes, ma'am, done. Thank you. I would like to thank KC College and my guide. Yes, I'm out open for questions.
Okay. Does anybody have any questions? Okay, if not, I wish you good luck for your future correlation studies with the soil quality and the properties of the plant and the medicinal properties. Thank you very much, Sharon and Dr. Damle, both of you. We will move on to the next speaker, which is probably Kelly Nigrel.
Yes, sir. Give me a minute. I'll just share my screen. Yeah, is the screen visible?
Yeah, now it's visible. You can start.
Okay, good afternoon. I'm Kelly Nigrel and I'll be presenting our research study titled A Survey-Based Analysis of Available Treatments Used by the General Populace for the Management of an Acne Infection. Acne is a common skin infection that has been the 8th most frequently occurring infection globally with a prevalence of 9.4%. The peak incidence rates of acne vulgaris have been observed in adolescents, especially between the ages of 14 to 19 years. Even adults and young children are susceptible to this infection. So acne is characterized by the development of skin inflammations on various parts of the body like the face, neck, back, chest, and shoulders. There are several factors that can lead to an acne infection, such as hyperseborrhea, hormone imbalance, or follicular hyperkeratinization. And all these factors create an environment conducive for acne-causing bacteria to thrive, leading to the onset of the inflammatory process. So as the severity of infection transitions from a mild to a severe infection, the normal pore becomes a microcomedon, and then comedons like whiteheads and blackheads, and then finally to inflammatory lesions like papules and pustules as shown in the figure alongside. Why is there a need for alternative treatments? Acne can affect the quality of life. It has a negative socio-psychological impact. People suffering from acne show symptoms of depression, anxiety, and low self-esteem even when they suffer from mild to moderate infections. Though the existing allopathic treatments, hormonal treatments, laser therapy, etc. are effective, they have several drawbacks. The major disadvantage of using topical or oral antibiotics is the development of multi-drug resistance infections. Additionally, there are several side effects of these drugs. For example, topical retinoids stimulate localized irritation, resulting in erythema, dryness, peeling, scaling, mood swing, etc. The hormonal therapies and laser treatments can be expensive for common people. And the hormonal therapies are also known to induce mood swings, nausea, and vomiting. The commercially available cosmetic treatments seem to be more about advertisements and actually a cure for the infection. Therefore, traditional, cost-effective, and safer alternatives are in need of the hour. There are several research studies that show the effectiveness of Ayurvedic and herbal remedies in the treatment of acne. Plant-based oils, leaf extracts, paste, etc. have synergistic properties useful in dealing with an acne infection. In this current research study, we have assessed the preferences of the general population with respect to 3 categories of treatments. The first being allopathic treatments that involve synthetic chemical drugs. We considered both topical treatments and oral antibiotics. Herbal or home remedies, which included plant-based treatments and simple or home remedies like applying an ice pack. And then we looked into commercially available cosmetic products, which included cosmetic and healthcare products like face washes, scrubs, or creams that are commercially sold in the market. Through our survey, we collected data on the basis of the following parameters: severity of infection across the age group, the preferred type of treatment used to cure acne, the relationship between the severity of infection and type of treatment used, the effectivity of these treatments according to our respondents, the most commonly used treatment in each of the three categories, the role of advertisements in the choice of treatments, and lastly, the respondents' willingness to use herbal remedies. Moving on to the methodology, this was a survey-based research project, so we created questionnaire using Google Forms. We ensured to include questions that were short and simple to understand. The conditional logic was used so that respondents could answer questions that were only pertaining to them. Both close-ended and open-ended questions were included. Additional explanations were provided wherever necessary so that participants better understand and respond to the questions asked. The survey was also approved by a professional in the field. The target population for the survey included a randomized pool of individuals, both males and females, between the ages of 14 to 55. We conducted a trial run with 12 participants to ascertain that the questions were framed in a way that the participants could answer appropriately. After making necessary revisions, we carried out a pilot study with 95 participants and a main study with 444 participants. In order to distribute our survey form, we sent out the flyer shown below through online social media platforms. The flyer contained a brief information about the project and the Google form link of the questionnaire. We made sure to obtain the consent of the participants. The data we collected was stored in a passport-protected WinZip file. Confidentiality of individual identities was maintained throughout. For analysis, the data was segregated, tabulated, and statistically analyzed using Microsoft Excel 2019 and GraphPad Prism 5. Moving on to the results and discussion of the main study. The main study was conducted with a total of 444 participants. Firstly, I'll discuss the differences we observed across the gender. The combined line axis on the secondary axis shows that females reported a higher severity of infection than males. The clustered column chart on the right depicts that maximum participants get their first case of acne as adolescents, that is from the ages of 11 to 20 years. And then we also observed that from ages 11 to 15, a higher percentage of female respondents reported the onset of an skin infection as compared to males in the same age group. While in the ages of 16 to 20, more males than females experienced their first symptoms of acne. Therefore, it can be said that females reported an early onset of the infection. This slide highlights the treatment preferences. So we divided our participants into three age categories, 14 to 19, 20 to 24, and 25 above. Analysis was done in accordance to these categories. The box on the top right provides a count for each category. This compound graph plots the total participants choosing allopathic treatments, herbal and home remedies, and commercially available cosmetic products. It's observed that 76.36% of the total surveyed population have used herbal and home remedies as a method to cure their acne infection, and a similar trend follows across the three age groups. Further, a total of 128 participants use allopathic treatments. These medications were the least form of treatment used. Therefore, there were maximum users for herbal and home remedies, followed by commercially available products, and then allopathic medications. Only 11% of the total surveyed population chose not to use any form of treatment against their acne, which could be attributed to their low severity of infection. Coming to the severity of infection, this radar chart illustrates that the average severity of infection of all 444 participants was 2.097 on a 5-point Likert scale. It's observed that with increase in age, the severity of infection decreases. The maximum severity of infection was observed between ages of 14 to 19. The mean severity of this age group is higher than that of the total population surveyed. This bar graph associates the treatment preferences with the severity of infection. People with less severe infection preferred to use home remedies, and as the severity increased, they started purchasing commercially available products and eventually started relying on allopathic medicines. Those with the most severe infections preferred allopathic treatments. So here, participants scored the effectivity of allopathic treatments, herbal and home remedies, and commercially available cosmetic products on a 5-point Likert scale. We observed that the mean effectivity scores of synthetically manufactured allopathic treatments and simple home remedies were comparable to each other, while that of commercially available cosmetic products had a considerably low effectivity scores. So to further ascertain these results, we carried out an ANOVA using GraphPad Prism 5 followed by Tukey's multiple comparison test, where each group was compared with every other group, and this test is carried out as a post-hoc after ANOVA. Here at a p-value less than 0.05, it was observed that there was no significant difference between the average effectiveness of allopathic versus herbal treatments, while there was a significant difference observed between both the allopathic versus commercial as well as herbal versus commercial. Therefore, the results show that the efficacy of allopathic and herbal treatments are nearly the same, while these two types of treatments are superior in curing acne than commercially available cosmetic products. Out of the 444 participants surveyed, 82 respondents used all the three types of treatments, And evaluating the data of these 82 respondents showed that they recorded the highest mean severity of infection, higher than the average severity of the 444 participants surveyed, as seen in the graph alongside. This suggests that the increased severity of infection in these 82 encouraged them to utilize most of the available treatments. The average effectiveness rating of these 82 participants for allopathic medicines was
Ma'am,
the
you
highest.
have 2 minutes left.
Yes, at 3.17 on a 5-point Likert scale. However, more than 50% of them experienced side effects like itchiness, skin reddening, etc. after using allopathic treatments, and therefore, when asked, 97.56% of them were willing to use herbal treatments in the future. To speak about the influence of various parameters on the choice of treatments, the inner ring we represented a donut chart, and the inner ring represents herbal remedies, and the outer ring represents commercially available cosmetic products. It is seen that advertisements played a major role in promoting the commercially available cosmetic products as compared to herbal remedies, while YouTube and word of mouth played a major role in promoting the herbal remedies. The respondents were also asked to rate their willingness of using herbal or Ayurvedic remedies in the future, and they rated a willingness of 3.81 on a 5-point Likert scale. We even determined which product was most commonly used in each of the 3 categories. In allopathic treatments, topical treatments were most preferred and over oral antibiotics, and the most commonly used ones were benzoyl peroxide, salicylic acid, and clindamycin. In herbal remedies, aloe vera and turmeric were most preferred, while in commercially available cosmetic products, Himalaya and Clean and Clear were the most used brands. To conclude, we see a need to assure access to affordable and safe quality medicines to combat with acne infections. Our research project finds that people preferred to use allopathic treatments when the infection was severe. However, the results also show that allopathic medicines may not be the best option for the holistic management of acne, as they induce side effects. 46.1% of the participants using these treatments have reported side effects. Moreover, the efficacy of herbal treatments as provided in the survey statistically shows that herbal treatments are at par with allopathic medications. Herbal and home remedies have been shown to be effective in reducing acne inflammations, and the results also highlight that only 6% of herbal remedy users took the guidance of an Ayurvedic practitioner. There are many plants. Yeah, I'll just take a minute.
Please try to conclude.
Yeah. There are many plant-based preparations that have anti-inflammatory properties or inhibitory effects on the growth of bacteria. However, the allopathic method is more scientifically standardized and more regulated. For this reason, these chemical drugs still seem to be the choice of treatment in severe acne infections. Lastly, commercially available cosmetic products have least effectivity scores, which suggest that they may lack the necessary curative properties to cure the acne infection. These are marketed as cosmetics and not as pharmaceutical products, and their popularity can be attributed to the marketing prowess of the big banner products. These are our future prospects, where we want to assess the validity of commonly used remedies in a multi-stage microbial assay to a certain dosage standardization for the treatments, and we also wish to scientifically promote the use of herbal and Ayurvedic medicines. These are our references, and we're extremely grateful for the organizers for giving us an opportunity to present our research. Thank you.
Okay, thank you, Kelly. Because you went over time, we will not go for any questions right now. We will move to the next speaker.
Okay, sir. Thank you.
Can I request Aryaman Tulpule to come online?
Yes, sir. Am I audible?
Yes. Please share your screen and can you start?
Yes, sir. One second.
He will tell us about microbial analysis of the Banganga water tank.
Yes, sir. Is my screen visible?
Yeah.
Okay. Good afternoon, everyone. I, Ariman Tulpule, a TYBSc student, will be presenting a research project titled a Physicochemical and Microbial Analysis of the Banganga water tank. So Banganga tank is an urban freshwater pond located in Malabar Hill, Mumbai, and is rich in its religious and historical significance. There are numerous temples and dharmashalas that are situated around the tank as if guarding the sanctity of the pond. Delving into Indian mythology, it is believed that when Lord Rama was traversing this land on his way to Lanka to save Sita Mata, he shot an arrow into the ground, causing water to sprout and quenching his thirst. Even today, there is a pole in the pond signifying the spot where the arrow struck. The pond is witness to daily religious rituals like Asti Visarjan, and once a year is an official immersion place for idols. Upon learning about Banganga's deep history and religious importance, we felt it was essential to visit the site and experience this for ourselves. Recently, we had a degree college excursion to Banganga tank, and in our minds, we had built up the image of a pristine, holy location, but what we witnessed was in stark contrast to what we had envisioned. When we arrived at the pond, we were appalled at the conditions of the tank and its premises. Various anthropogenic activities were being conducted in and around the tank, and locals were seen bathing in the pond while some were even washing their clothes, and leftover food and offerings from nearby temples were strewn along the banks. Ducks, crows, and swans were all seen eating the vegetables and other waste that was left on the steps. And one of the local residents there told us that sewage that flows through the gutters under some of the steps was also seeping into the tank, which can be observed on the top right-hand side of your screen in the photo. Looking at this, we asked the responsible personnel how often the tank is cleaned and was stunned to find out that the pond is drained of water only once every year for cleaning purposes. So the debris are cleared, but what about the condition of the water? To this, the person further went on to tell us that there is no treatment done to the water of the tank, which led us to believe that the people were unaware about the actual state of the water. In the photo on the bottom right of your screen, the man is observed to be fishing in the tank. That is when we realized that the people would probably be eating fishes from this pond for quite a long time now, and the potential implications of this on their health are unthinkable. So we were also told that during the summer months of April and May, there are even swimming lessons for children being conducted in the pond. So imagine the degree of bioaccumulation and eventual biomagnification of all the probable contaminants present in the pond due to the prolonged exposure and their probable long-term effects in the children and the adults who use this pond for recreational activities. As is visible on screen, there were plastic wrappers, bottles, and containers all floating on the surface of the water, along with domestic waste being thrown into the water. After being completely disillusioned, we decided to find out more about the situation. And while conducting preliminary research, we came across several newspaper articles that corroborated to what we had observed. It was unfortunate, but as we researched, we found out the scenario isn't new at all. It has been so for a long time, as this 2011 article in the Times of India suggests, that the degrading conditions of the water result in declining numbers of ducks, swans, and tortoises, all that inhabit the pond. 6 years later, and the problem of death of fish still persisted. This 2017 article in the Hindustan Times reiterates the fact that dwindling numbers of fish in the pond is because of the issue of overfeeding, and that the trust that owns and maintains the tank removes up to 1 ton, I repeat, 1 ton of dead fish from the waters every year. Another article published in 2020 from the Times of India reported that the water of Banganga was turning murky due to construction activities ongoing in its near vicinity, threatening to permanently damage the natural aquifer that supplies water to the pond. The construction work and its effect on the pond forced the mayor into action, as reported by this 2021 article published in the Deccan Herald, who ordered the work to be stopped with immediate effect until further assessment could be conducted. So the articles we collected from 2011 to as recent as 2021 all highlight the continually neglected condition of the tank, yet no work has been done to reinstate the original conditions. Fish and other inhabitants of the pond continue to perish year after year, yet no solution is forthcoming. As you can see, the evidence emphasizing the dire state of the pond and its ecosystem is alarming. Intervention from the mayor has had no tangible impact on the condition of Banganga. Even today, the water continues to be murky, fish continue to die, and anthropogenic activities continue to contribute to the depleting conditions of the water. Witnessing such a calamitous scenario, we felt it was imperative to conduct an analysis of the tank ourselves to better understand the current conditions of the pond and to showcase the deteriorating state of the water. So the aim of a research project was to perform a physicochemical and microbial analysis on the samples collected from the tank, to assess the quality of the water of Banganga tank, to identify its aquatic microflora using microscopy, and to create awareness regarding the degrading conditions of the tank. To begin with, we collected samples from various areas of the pond, and the temperature of the water was recorded to be 24 degrees Celsius. The samples were collected in sterile containers and sealed with paraffin sealing film before being immediately transported back to the lab. The samples were then analyzed using standard methods outlined by the American Public Health Association in triplicates, and the result obtained from the samples were compared to acceptable limits set by the Central Pollution Control Board and the Bureau of Indian Standards for drinking water. Analysis of the samples was completed on the same day of collection. The samples were also analyzed for the presence of microflora using microscopy, and the results encouraged us to further conduct a microbial analysis. So the results of a physicochemical analysis were as follows. The pH for both the control and test sample was measured using a pH meter, and the pH of the test sample was found to be 7.37, while the acceptable limits set by the Bureau of Indian Standards say that the pH should be within the range of 6.5 to 8.5. However, the case was not so with the other parameters we analyzed. The next parameter was total hardness, and total hardness was calculated to be 80.6 milligrams of calcium ions per liter, which exceeded the acceptable limits, which were 75 milligrams of calcium ions per liter. Evidence of the construction work around the pond, as suggested by the newspaper articles I mentioned before, can be seen in the incredibly high total hardness value of our test sample as compared to the control, which was just 3.2 milligrams of calcium ions per liter. Calcium ions present in the cement and concrete used for construction may have leached into the pond, resulting in the severe contamination. Another critical parameter is chlorinity, and its levels were observed to be extremely high at 698.64 milligrams per liter as compared to the acceptable limit set by the BIS at 250 milligrams per liter. The values were reconfirmed by analyzing and resampling in triplicates. Elevated levels of chlorine could be the result of increased sewage contamination that was observed to be seeping through the cracks in the steps around the tank. And this also supports the algal blooms that were observed in the pond. Alkalinity of the test sample at 125 milligrams per liter was observed to be within the acceptable limits set at 200 milligrams per liter. However, compared to control value, which is 55 milligrams per liter, the value of the alkalinity of our sample is very high, which can be again attributed to introduction of anions such as carbonates from construction work being done in the vicinity of the pond and through domestic activities that were observed on site. The observed alkalinity can also be related to the elevated levels of total hardness of a sample. The limit set by the CPCB for DO said that the minimum amount of dissolved oxygen in the test sample should be 4 milligrams per liter or more. Our test sample contained a very high concentration of dissolved oxygen at 22.7 milligrams per liter. Further, it was observed that the BOD value of the sample was 5.41 milligrams per liter, whereas the acceptable limit suggests that BOD of water should be 3 milligrams per liter or less. A higher demand for oxygen in a test sample suggests that there is a presence of microorganisms that need oxygen to break down organic matter present in the pond water. And this also suggests that the water of Banganga is contaminated and is clearly eutrophic. So to identify the microflora present in the water samples, we performed microscopy, and there were numerous observations made in the water, but here we are just highlighting a few for the sake of reference. So these are liposyncylis, which are green flagellates belong to the family Fasaceae and are mainly found in freshwater habitats that are rich in organic matter. Another such example is the presence of Pediastrum, which is a genus of green algae that are part of freshwater plankton, and Spirogyra, commonly known as pond scum, is a green filamentous algae that is found all over the world in freshwater habitats and especially in eutrophic waters. These are certain diatoms that we observe, such as Cyclotella and Navicula, and certain species of Cyclotera are said to be resistant to extremely high polluted water bodies, whereas certain species of Navicula are said to be resistant to high levels of pollution. Here are a few videos showing some of the phytoplankton. The video on the left shows Navicula, which is a highly motile diatom, and the video on the right shows the species of Liposyncylis in action. As I mentioned earlier, that while visiting the tank, we had observed many people bathing in the water and the water being consumed as part of
We have
religious
2 minutes
ceremonies.
left, sir. Please try
Yes,
to
ma'am.
conclude.
So we were quite shocked to find Liposcylus, which is a genus of pale brown wingless insects that are associated with the freshwater habitat. And studies have shown that Liposcylus species have a potential to act as allergens and may result in strong skin sensitivity. And on the left is a picture showing that a man was bathing in the pond. So you can imagine the kind of health risks that people are exposed to every day. And the pictures on the screen, they show a clear indication of the amount of plastic contamination in the pond. And the photo on the left shows the presence of probable microplastics based on the literature available containing photographic records. Coming to microbial analysis, we performed an MPN test wherein we took 15 tubes in total, of which 5 were double-strength McConkey's broth, 10 tubes of single-strength broth containing 10 ml, 1 ml, and 0.1 ml of the samples were taken. These tubes were sealed and then incubated to see if discoloration occurred. And discoloration was observed if there is a presence of lactose fermenting coliforms that feed on the lactose in the broth to proliferate. The higher the discoloration of the tubes, the greater the contamination of the sample. So for our sample, all 5 test tubes containing double-strength McConkey's broth and single-strength broth incubated with 1 ml of sample showed discoloration as compared to 4 tubes changing color, which had only 0.1 ml of the sample. The tube on the right shows the control, which was just 10 ml of McConkey's broth. So the results show a very high number of colony-forming units in sample 1 at 1600 CFU per ml, followed by a similar showing in samples 2 and 3 at 900 CFU per ml. So the results were compared with an MPN index, and it shows that the extremely poor quality of the water body due to presence of coliforms, and such a high number of bacteria also attributes to the elevated BOD values that were observed during our physicochemical analysis. This is Saboras dextrose agar, which allows the growth of saprophytic fungi, and our primary identification revealed probable colonies of Rhizopus aspergillus or yeast in the water sample. Similarly, results for tea sticking showed a vast amount of bacterial growth in nutrient and McConkey's agar plates, and preliminary identification revealed probable colonies of gram-negative E. coli or enterobacteria.
So your time
So
is
I'd like to conclude that the results that have been showed so far highlight the dire condition of the pond. These activities have been continuing unmonitored and led to the severe degradation of the water quality, and elevated levels of certain parameters that we observed than the standard values indicate that the water is non-potable and non-usable. Further, it poses a serious threat to human and aquatic health due to the presence of algal blooms, coliforms, fungal and insect growth that was found in the water. Despite the visible degradation of water quality, there have been no measures being taken to reverse the situation. And there are no official reports on the water quality assessment of Banganga in the public domain. So there is a need for quarterly cleanup and sanitization of the water and its premises. and the scientific-driven government intervention is a necessity for maintenance and restoration. In terms of future prospects, we are working towards raising awareness within the local residents as well as the general public. We are also
Sir,
continuing
please
our study with...
Time is
Yes,
already
I'm
running
just
up, sir.
finishing. This is
Please
last
continue.
slide. Such as heavy metal detection, we're also testing the water during seasonal variations and after festivals. We are in the process of continuous microbial analysis to identify the type of bacterial and fungal contamination in the water sample. And we would also like to join hands with NGOs to help in their restoration drives and start making a visible difference. And finally, we are planning to conduct assessment of more such freshwater ponds in the future. These are the references. I would like to thank
thank you.
Yes, sir.
Thank you, Ariman. You gave a very comprehensive study of Banganga, and I wish you all the best for following up on this and hopefully having a Banganga tank which is cleaner and safer, and at other places also. Thank you very much.
Thank you so much, sir.
We will not take questions because we are running out of time now, and we will move to Vaidya Rakesh Salve for the next talk.
Yes, good afternoon, everybody. Sir, am I audible?
Yeah, please share your screen and get started.
Yes. Yes, are the slides visible, sir?
Yes, please start.
Yeah. First of all, I would like to thank the organizers of the International Virtual Conference on Life Science Research and its interface with Allied Sciences 2022. Myself, Vaidya Rakesh Salve, currently working as a professor in Arihant Ayurvedic Medical College and Research Institute, Gandhinagar, Gujarat. My topic of today's presentation is biological development mentioned in Ayurveda. Can epigenetics put some light?
Hello.
Yes, sir. Sir, I'm not able to change the slides, so just a second, sir. Sir,
are you sharing your entire screen or just one window?
No, I mean, I'm sharing the entire screen, sir, entire screen.
Okay, go out and then again start sharing.
Just a second, sir. Sharing entire screen. Yeah. Are they visible, sir?
They are visible. Go into slideshow and let's see. It's changing. Yeah, go to the next slide. Yeah, go ahead.
Yeah, thank you. So first of all, maybe you must be wondering why am I participating in this seminar. Referring to a very popular reference from Acharya Sushruta Samhita, where Sushrutacharya says ki ekam shastram adhiyano na vidya shastra nishchayam tasmat bahushrutaha shastram vijaniya chikitsakaha. Reading only one science may not give us sufficient knowledge. Hence, one should read different sciences having the same basic principle or goal. This enables us to become ideal clinicians. The Human Genome Project. Everybody must be aware of this audacious project, which was an attempt by the human being to crack down the life's code. It was a 13-year-long publicly funded project initiated in 1990 with the objective of determining the human DNA sequence. The result of this project yielded us a mosaic, which does not represent any one individual and was declared complete in 2003. Now, friends, generally we decide or we consider DNA as the game decider. However, when we see the result of the Human Genome Project, the difference between human genome and the chimpanzee genome ranges only from 1 to 4%. So being so close to the chimpanzees, we look, act, think, as well as use our skills very differently. The functional unit of the DNA are genes encoded which behave according to the signals received by them. The gene expression thus is the deciding factor for all these differences. The gene expression depends upon a number of factors, that is internal, that is inside the body, inside the human body, as well as the external, that is the external environment. The experiments related to probing of the human DNA some years back has shown changes which are attributed by its exposure to stress conditions like UV rays, drugs, foods, especially food colors also. Thus, the bridge between the genome and the external factors may be the epigenome, which can be studied in the form of epigenetics. The word epi here means upon, on, top, or beyond, while genetics is related to the genes, genetic code, or our genes. Thus, the factors which affect the gene expressions can be summarized in this figure, where the genome is surrounded by the epigenetics, that is the bridge between the external environment as well as the genome. The external environment, that is the environment in which a person grows, or maybe he or she is conceived in the womb of her mother, also has an effect on the final gene expression. The economic factors under which a person is growing, the drugs, the medicines to which a person is exposed right from the antenatal period up to the postnatal midder, as well as in its early days of growth. The age to which a person belongs also has a direct effect on the gene expression. The food, as we can all see, we have different food habits in different parts of our own country. So the food to which a person is exposed in its early life also has a profound effect on the expression of the gene. The social factors, the gender also has a profound effect on our gene expression. An interesting study which was published in International Journal of Epidemiology way back in 1999, I have took some conclusions from the paper. They conclude as the past decade result has suggested that prenatal and early postnatal nutrition influence the risk of developing chronic degenerative diseases up to 60 years later. And that is a very big time. Young adulthood death from infectious diseases is similarly programmed by early life events. Early life exposures correlated with season of birth strongly influence susceptibility of fatal infections in young childhood. Nutritionally mediated intrauterine growth retardation may also permanently impair development of our immune system. Now, friends, coming to our Ayurvedic part. When we see the Ayurvedic factors which are considered essential for conception, again we can see the reference from Sushruta Samhita as Dhruvam Chaturnam Sanidhyat Garbhasat Vidhi Purvakaha Rutu Kshetrambu Bijanam Samagrat Ankuroyatha. Now, Acharya Sushruta has used the simile of agriculture here. He enlists the four important factors which are important for ideal agriculture. He says season, land, water, as well as seeds. These are the four factors which are important for ideal agriculture. The same four factors, when converted into human biological factors, can be considered as season, which is correlated with the ovulation period, that is Rajas Samayaha. Land, which is the agricultural land, can be correlated with a healthy uterus, which the female should have before going for conception. The water, which is important for agriculture, can be compared with the food taken. And food here is not only solid food, but solid as well as liquid food, or the nutrients which are absorbed from the mother's blood, which are responsible for the ideal growth of the fetus. And seeds, the most important factor, that is the ideal male and female gamete, which are finally responsible for ideal conception, implantation, and growth of the human body. One interesting concept of Ayurveda is the Prakriti, that is the human constitution. And when we see the determining factors which are responsible for human constitution, they are considered as three doshas, Vata, Pitta, and Kapha. And if you see the ideal characteristics, just to enlist some of the characters, if you see the Vata constitution, he must be having less hair. In Pitta Prakriti, it has early graying of hair, and in Kapha Prakriti, they have lustrous hair. The build of Vata Prakriti is thin, Pitta Prakriti is thin or medium, while it is more towards the obese side in a Kapha Prakriti. Similarly, skin, speech, mind, and my friends, dreams also, that acharyas have enlisted different dreams according to the different constitution. Now I'm coming to the Vikruti, that is the diseases. The congenital characteristics or diseases also witness are decided by the doshic predominance. For example, a person with Vata predominance generally has hearing problem. He may produce dumb if Vata is more during the preconception period. He may have a voice which is nasal or hoarse in nature. He may be born in the blind condition, or he may be dwarf in his height. In a pitta predominance as well as kapha predominance, there are different conditions. Just to enlist some, baldness or graying is more in pitta prakriti, vitiligo is more in kapha predominance, congenital teeth may be present in a kapha predominance child. And hence, if you see the Ayurvedic text, they suggest ideal food or behavior as well as ideal medicines to
Sir,
be
2 minutes left.
yes,
Last
to be to be administered to the pregnant lady so that it may promote ideal growth of the fetus as well as normal delivery also. And similar to the ideal food behavior, harmful food and behavior or harmful effects are also suggested. For example, being oversleepy. If the pregnant lady is oversleepy, the child may take birth with a sleepy nature, as well as it can have some less digestive power. If the pregnant lady is exposed to more alcohol in the ANC phase, the baby may take birth with overthirst as well as short memory. These are some of the examples. Thus, the detailed references, if we see Ayurveda, you can have poor proper age of conception, formation of fetus with 6 components, vital nourishment, month-wise development of fetus, month-wise changes in the pregnant female, factors deciding the complexion of also of the baby, organ genesis, as well as management of the puerperal woman. These all are explained in quite detail in the Ayurvedic text. So my final words should be Ayurvedic Samhitas, that is texts, have a detailed description of prenatal, antenatal, as well as postnatal changes happening in the female body. They describe detailed growth and development of fetus. Moreover, the parts of fetus to be developed in particular months has also been described. The prerequisites needed for the development of healthy heart, liver, spleen, intestines are also explained. Epigenetic studies may prove useful or very useful in understanding the relevance and application of such huge scientific information which is explained in Ayurvedic texts. Thank you. I once again thank the organizers for giving me the opportunity of presenting my thoughts on epigenetics or application of epigenetics in organ development described in Ayurveda. Thank you.
Okay. Thank you, Dr. Salve. Are there any questions related to this talk? Anybody has a question? Okay, if not, we will move on because we are short of time now. Thank you, Dr. Salve. Now we will move to behavioral science actually, and we have Dr. Sunita Girish Pimpale to tell us about irrational financial decisions.
Yes, sir.
Yes, can you share your screen?
Yes, sir. Just a minute, sir. Am I audible, sir?
Yeah, we can hear you and we can see your screen also. Please start.
Yes. I'm Dr. Sunita Pimpe, Head Department of Costing from BYK College of Commerce. At the outset, I thank the organizers for giving me this opportunity. The title of my paper is Irrational Financial Decisions, a Reality. I know you all must be wondering that how
So you have to go into slideshow mode because we can hardly see anything what is written on your slide.
slideshow mode. Sir, this is the first slide I am showing.
Yes, but you are not in the slideshow mode, so your slide is really small. We cannot read anything.
Okay. So what should I do, sir?
Now we have option for slideshow on the top.
Okay.
File, home, insert, design, transition, animations, and slideshow. You can click on slideshow.
Is it okay?
No, ma'am.
No,
Is this
at the top?
Yeah, click that.
Okay,
Yeah,
yes,
click that.
Click that.
Yeah.
Is that okay, sir?
No, nothing happened. Maybe you
You
can
can go
just.
at the top, ma'am.
Okay, just a minute.
You can see file. Yes.
File, yes.
Yes. Home, insert, design, transitions in the same sequence.
Insert, design,
Transition,
transition, animation, slideshow. Okay.
From beginning.
From beginning. From beginning. Go to the left. From beginning.
Okay, yes, okay, yes. Got it. Is that okay?
No, I think there's some problem with your this thing. So just why don't you just continue? We will hear you. Yeah.
Okay. I know you all must be wondering that how financial decisions are linked with life sciences. But research has proved that there are biological reasons that push us towards certain psychological biases. Within each individual, it is the pattern of various hormones. These hormones make changes and that determines the influence they will have on financial decisions. That's the reason I am here in the International Conference of Life Sciences. For this presentation, we need to know some basics of economics and behavioral finance, and then we'll turn to financial decisions. And that's why we'll first see the rational choice theory. Humans are rational actors. Decisions they take are based on cost-benefit analysis. Rational decisions are taken by effectively weighing the cost and benefits of each option available to them. The financial decision made will be the best choice for the individual. The theory believes that rational person has self-control and is not moved by emotions and external factors. Now we'll see the rational decision-making process. Rational decision-making process according to rational choice theory starts with the identification of an opportunity. Once the problem or opportunity is identified, people gather all information related to opportunity. With this information, they develop the options for the decision-making process. The rational choice theory believes that after evaluating various options, people select preferred alternative rationally. Next comes the behavioral economics. It studies the effects of psychological, cognitive, emotional, cultural, and social factors on the decisions of individuals and institutions. In simple words,
excuse me, ma'am, please change. Are you changing the slide?
Yes, I am changing the slides.
Your next slides are not visible, ma'am. Please click on slide 2.
Next slide?
No, the next slide is not visible. From left hand side, you can change it.
Is that okay?
You can change it sequence wise.
Yes, I have changed the sequence wise. Now we are in behavioral economics. That's why it is a slide number 5.
No, it's
so
not.
So can you go out, just, go out from sharing and share your screen again?
Okay.
शेअर करायचं आहे ते सिलेक्ट करून मग ठेव.
Is this okay?
Yeah, now go into slideshow again.
Yes. Is this okay, sir?
No, it's not. I think you just continue with your talk because there's some problem which we won't be able to fix in such a short time. Just go ahead, continue your talk.
Okay. Now about the behavioral economics. That explains economic analysis that applies psychological insights into human behavior to explain economic decision making is nothing but behavioral economics. As we are discussing here the financial decisions, we need to know about behavioral finance. So behavioral finance. Behavioral finance is of recent origin. Nobel Prize winner Daniel Kahneman is one of the founding fathers of behavioral finance. Interestingly, he and his friend Amos were both psychologists with no training of classical finance. Every person takes number of financial decisions in his personal and professional life. As a consultant, decisions regarding others' money are also taken. Behavioral finance is the study of the effects of psychology on investors and financial markets. It focuses on explaining why investors often appear to lack self-control, act against their own best interest, and make decisions based on personal biases instead of facts. Humans are normal and cannot be always be rational. People are also influenced by their own biases. They make cognitive errors. People have limits to their self-control. This results in irrational financial decisions. As the next slide says, irrational behavior is the result of cognitive bias, herding effect, lack of control, and present bias. The first is cognitive bias. Cognitive errors result from incomplete information or the inability to analyze the information that is available. Cognitive biases can affect person's decision-making skills, limit problem-solving abilities, hamper career successes, damage the reliability of person's memory. Sometimes people have incomplete information and sometimes they are unable to analyze the available information. In case of cognitive bias, financial decisions are based on irrational assumptions. When irrational financial decisions are the result of crowd psychology, it is called as herding effect. For example, use of popular brand. There may be less expensive brand with greater utility value, satisfying particular customers' requirement. Still, many times we buy expensive brand. Once a person loses control over the consumption, he starts taking irrational financial decisions. There are many short-term pleasures which spoil our life. Visiting expensive restaurants frequently just for a pleasure can be an example of this type. We can find number of examples where people give preferences to short-term pleasures, ignoring their long-term costs. Knowledge of behavioral finance helps an investor to see flaws in the thinking of other investors and to avoid those flaws themselves. The recent pandemic, COVID-19, has also troubled the world. World stock markets are again beaten down. This also opens up many opportunities to buy quality stocks at low prices. But people who are risk averse resort to safe assets like gold. Due to the massive buying boom of golds, the prices of these assets, that is gold or US dollar, increased a lot. And that of other, that is quality stocks, decreased. Because people pull out their money from other assets to invest in gold. This is a type of irrational financial decision. The financial crisis in 2008 and 2009 led many investors to exit from the stock market. Many had miserable view of the markets and expected more economic hardship in coming years. The experience of having gone through such a negative event increased their bias that the event could reoccur. But in reality, the economy recovered and the market bounced back in the years to come. The rational investor should buy when stock prices are dropping and sell when they are booming. If an investor has not understood the behavioral aspect of finance, they too are likely to blindly follow the trend. This means that they are likely to sell when the markets are crashing and buy when they are booming. Our day-to-day lives are full of such behavior. A common example of such decision is credit card versus paper money. For the same amount of payment, we take more pains by using credit cards. Another example of irrational financial behavior is buying lottery ticket with a 1 in a million chance of winning. We find n number of examples of irrational financial decisions in daily life of a common man also. These can be buying something which you do not afford. This happens particularly when a person wants to impress someone else. Sometimes we buy luxury items when money has been kept for necessities. We buy something unnecessary simply because it is on sale. Sometimes we put extra thousands of rupees on the credit card to just earn 100 rupees bonus.
Ma'am, you have 2 minutes left.
Yes. A general reason for our irrational money decisions is that we are simply emotional creatures. We don't always do what's best for us. It has been observed that most people have texted the messages while driving, though we all recognize its danger. Same is applicable to financial decisions. In the real world, the majority of people make important financial decisions more casually while at the dinner table. Thank you for patient hearing. Session is open for question and answers.
Okay, thank you, Dr. Pimpale. Do people have any questions on risk taking and irrational decisions? No, they don't. Okay, so we will conclude. Thank you, Dr. Pimpale. We move to the next one, which is YD Harita and Dr. Bilkis, who will tell us about study on the personality of adolescents.
Yes, sir. Thank you, sir.
Dr. Pimpale, can you unshare your screen? Stop sharing your screen. Go to the bottom, stop sharing your screen. Go to that arrow sign at the bottom. Yeah, can we have Harita share her screen?
Yes, sir. Yes, sir.
Is that okay, sir?
Yeah, that's fine. Thank you.
Okay.
Sir, is it visible, sir?
Yeah, I can see your window, but I can't see your PowerPoint.
Okay, sir. Sir, now, sir?
No, not yet.
Not visible.
Are you sharing your screen again?
Yes, sir. Yes, sir. Sir, I'm clicking the button of share button, sir, but it's not actually opening.
At the bottom, this arrow sign, are you present now? Are you clicking that?
Yes, sir. That's what, sir. I'm clicking the window button. And I'm trying to share the screen, sir, but it's actually not opening.
Okay, if you're not able to share your screen, then just tell us about what you have done looking at your own screen. We will just listen to you.
Yes, sir. Thank you very much, sir. Very good afternoon to the delegates and friends present in the conference panel. I am YD Harita, Department of Human Development and Family Studies from Home Science College, Acharya N.G. Ranga Agricultural University from Andhra Pradesh. The topic I'm going to present is on positive youth development, study on personality of adolescents. So generally, why I have taken only this topic means because youth is actually one of the prime factors, and where they actually in the stage like self-focused or economic or establishing stages, and yeah. So coming to the talking about the abstract, positive youth development is nothing but positive experiences and positive relationships, positive environments is called as positive youth development. And the positive developmental settings and the characteristics of successful positive youth development members can also be used for training staff, designing programs, and developing standards and assessment tools. That is physical and psychological safety, the safe and health promoting facilities practice that increases safe peer group interaction and decreases unsafe or confrontational peer interactions, and the appropriate structure, that is limit setting clear and consistent rules and expectations, firm enough control, continuity and predictability actually clears the boundaries, and age appropriate monitoring. The overall the results which I have found in my study is women reported highest score in agreeableness, neuroticism, and men scored highest in extraversion, consciousness and openness. So talking about the introduction part, that is the positive youth development is generally is a concept and practice of growing from dissatisfaction with a predominant view that underestimated the true capacities of young people by focusing on their deficits rather than their developmental potential. And positive youth development is based on the belief that given guidance and support from caring adults, all the youth which actually can grow up healthy and happy and making positive contributions to their families, schools and communities. And the term positive youth development often generates several different responses. Most commonly, people hear youth development and think of a child and adolescent development. The meaning is biological, social, emotional, and cognitive development, or they think of youth services, all the programming and services community provide to young people to address their needs and foster their growth, or they might think of a distinct approach or philosophy of interacting and working with young people, one that informs programming and promoting adolescent development. And it actually understood that positive youth development is an approach or philosophy that guides communities in the way that they organize services, supports and opportunities, so that all young people can develop their full potential. And the main thing is positive youth development is actually originated from ecological system theory, which actually focus on the strengths of the adolescents, and it is also similar conceptually with the principles of positive psychology that is central to its philosophy. The theory of positive youth development suggests that if young people have mutually beneficial relations with the people and the institutions of their social world, they will be on the way to a hopeful future marked by positive contributions to self, family, community, and civil society. So positive youth development, the establishment of sound positive environment around youths is a key to their effective functioning and survival. And positive environment is dependent on positive relationships and strengths established at school, family, or social environment through interactions with parents, teachers, peers, and society. And the development of positive ethos within youth is the foundation for better opportunities to understand themselves as youth and engage in the activities that would lead physical, psychological, and social upliftment of the youth. Positive youth development will actually enable youth to become successful learners, confident individuals, responsible citizens, and effective contribution. So talking about a study that is Flay Beer in 2002, he actually conducted a study and found comprehensive programs with positive results in multiple dimensions like that of risky, unhealthy, antisocial behaviors, poor mental health and poor academic achievement. And also Donald et al. In 2011, he also observed that study helps to better delineate relationships between community violence exposure and positive youth development by adding new knowledge to the literature on the role of family functioning. And the points of intervention should focus on families with attention to the parental figures in the home and overall family functioning. And Mueller in 2011, he also examined the relationship between adolescents' self-regulation skills and their participation in youth development programs across grades 8 and 9 in predicting grade 10 positive youth development and contribution. And the results indicated that self-regulation skills alone predicted positive youth development, and self-regulation and youth development program participation both predicted contribution. So talking so next is talking about on my research method. It actually focused on 11 to 24 years, and the main focus of the study was to study on positive youth development, study on personality of adolescents. And for findings, that is for research findings and discussion, I have used a scale of Big Five inventory tool, which actually includes areas or dimensions or extraversion, agreeableness, consciousness, neuroticism, and openness. So extraversion actually deals with sociable, forceful, energetic, adventurous, enthusiastic, warmth, etc. And in this area, 86% of the youth actually fallen in high category, and 8% of the youth fallen in average, and 6% were fallen in low category. And talking about the agreeableness, it actually deals with trust, trust or forgiving, straightforwardness, altruism, that is warm, compliance, not stubborn, modesty, and tender-mindedness, etc. In this area, 89% of the youth fallen in high category, and 7% of the youth fallen in
Stop, Pankaj.
average. And
Although I'm quite held up today in this meeting, it will take a 15 minutes.
तो मैं सेल्स लेने आया था
Okay.
कल मैंने डेक्सटान लाया था बट दिस स्टूडेंट सेल्स
shall I continue, sir?
Yeah, sorry, please continue. Continue.
Okay, sir. And 4% were okay, ma'am. And 4% were fallen in low category. And talking about third dimension or third factor, that is consciousness, which actually deals with competence, order, not careless achievement, striving, self-discipline, deliberation, etc. And in this area, 90% of the youth fallen in high category, and 6% of the youth fallen in average, and 4% were fallen in low category. And the fourth factor or fourth area is neuroticism, which actually deals with anxiety, angry hostility, depression, self-consciousness, impulsiveness, vulnerability, etc. And in this area, 83% of the youth fallen in high category, and 11% of the youth fallen in average, and 6% were fallen in low category. And talking about the last factor or last dimension, that is on openness, which actually deals with idea, fantasy, aesthetics, actions, feelings, values, etc. And in this area, 80% of the youth fallen in high category, and 12% of the youth fallen in average, and 8% were fallen in low category. So the final results I conclude is women reported high score in agreeableness, neuroticism, and whereas men scored highest in extraversions, consciousness, and openness. So the final conclusion I conclude is the data collected with respect to understand the study on positive youth development, study on personality of adolescents. The positive youth development motivates youth to achieve their full capacity and energies themselves for active engagement in meta development and the establishment of a sound positive environment around youth.
Ma'am, your time is up. Please continue.
Yeah, ma'am. Give me 30 seconds, ma'am. I'll wind up. So the development of a positive ethos within youth is the foundation for better opportunities to understand themselves as youth and engage in the activities that would lead to physical, psychological, and social empowerment of the youth. So thank you, ma'am. Thank you, sir.
Thank you. Thank you, Harita. I wish we could have seen your slides, but let's, do we have anybody has any questions for Harita? Okay, if not, we will now move to oceanography and to talks on
Excuse me,
oceanography.
sir.
Yes.
A bit change. The next paper will not be presented as the presenter is not ready. We'll directly switch on to the last presentation.
Okay, fine. That is ecological importance of copepods.
Yes.
All right, let's do that. But I thought it was the same presenter for both talks according to the schedule.
Yeah, but different presenters were there.
Okay, all right.
Hello, I am audible.
Yes, Mr. Islam, you are audible. Please share your screen.
Thank you, sir. Just a minute. Okay.
Yeah,
Is
please
it
start.
okay? Okay. Thank you, sir. Thank you. Hello, honorable professors, scholars. Good afternoon and warm greetings from Bangladesh. One thing is, we are the important component of Blue Planet, no doubt. This one is the most precious blessings from Almighty.
Can you go out? It's very small on the screen. Can you go out of this and go to the regular mode and zoom in? Because we can't read anything that you have.
Is it okay, sir? Is it
Yeah,
okay?
this is much better. Yeah, go on.
Okay, the things is we are the important component of Blue Planet. No doubt, this one is the most precious blessings from Almighty. As human being, we should keep our planet safe and comfort zone. So it's time to raise our hand, our voice, our brain, and our pen to find out the problems and their solutions to keep the Blue Planet better placement. Okay, sir. This is MD Saiful Islam from Noakhali Science and Technology University, Bangladesh. And first of all, I am very much grateful to the organizing panel to allow me for presenting my paper. Okay. Dear professors, I have worked on ecological importance of coffee foods, perspectives on their relation with diatoms and cyanobacteria in freshwater ecosystems. My research conducted under Biological Oceanography Lab, Department of Oceanography at Noakhali Science and Technology University. Now, let's see the introductions of my research. The freshwater ecosystems are interactive ecosystems in which biotic organisms, their growth and adaptability, as well as associated biological productivity and nutrient cycling are linked together surrounding. They are the most promising aquaculture sources. However, controlling the numerous complex abiotics and biotics elements that influence the effectiveness of fish farming is the most difficult task facing fish farmers. Controlling coffee food populations by modifying the pond environment, especially the abundance of diatoms and cyanobacteria concerning the physicochemical water parameters, is an illustration for the intricacy required in controlling aquatic systems. The next one is, there are a couple of objectives of my study. First one is monitoring the algal bloom in tropical water ponds to increase predictability of food fish productions. Second one is assessment on ecological importance of coffee foods in historical freshwater ponds. And the last one is to evaluate the relation between phytoplankton. Especially my research focus on diatoms and cyanobacteria with coffee foods. Now the method and materials. In conducting study, there are 3 historical freshwater ponds were chosen from Noakhali Sodor, Bangladesh. The present study covered 3 seasons between July 2019 to May 2020. And to measure the physicochemical water qualities, we also followed the American Public Health Association's 1995 to determining the physical water qualities. And we also measured dissolved oxygen, ammonia, alkalinity, free carbon dioxide, phosphate, temperature, transparency, and pH. Here look the study area map, and pond A, pond B, and pond C show the my chosen 3 historical ponds. Now let's see the result. According to my research, I have recorded 4 most dominant coffee food species from 3 ponds. They are the Neodiaptomas, Thermocyclops, Mesocyclops, and Heliocyclops, which are the most dominant species in freshwater ponds. And figures 1 shows that the plankton abundance in 3 ponds. According to these figures, we show that the abundance of cyanobacteria is higher than compared to diatoms and coffee foods. And among these, in pre-monsoon season and post-monsoon season in ponds B, we found the highest abundance of cyanobacteria, and second one is diatoms. They are the second most dominant species in three ponds. And figure 2 is showing the aquatic chemical parameters, which is covered with dissolved oxygen, ammonia, alkalinity, free carbon dioxide, and phosphate. Now, figure 3 showing the CCA biplot between environmental parameters with plankton genera, especially on diatom, Bacillariophyta and Copepoda in each pond, which is conducted with alkalinity, dissolved oxygen, free carbon dioxide, ammonia, phosphate, transparency, temperature, and some algal species such as Epanocapsa, Amphora, Epanotheci, Cyclothera, Plagiaularia, Navicula, Neodiaptomas, Oscillatoria, Synedro, Thermocyclops, and so on. Now the discussions of my research, the canonical correspondence analysis or CCA revealed inverse relationships among the 3 plankton groups, Bacillariophyta, Cyanophyta, and Copepus, as they occupied in the 3 different axes that is showing in figure 3. And the next one is the study investigated that cyanobacterial bloom have effect on copepod biomass because they secrete harmful algal toxins, especially cyanotoxins known as cyanotoxins, which are the unfavorable for copepod biomass. And the third one is, but there are a positive response of copepod to the diatom, but sometimes they can hamper the copepod biomass because diatoms, which are common primary producer in environment, but they are able to produce oxylipin, which have a negative influence on fitness or copepod of offspring. So the things is we have to control cyanobacteria bloom and also control diatom bloom to keep the environment friendly and for aquaculture purposes. And the conclusion is the monsoonal ponds provides an ecological purpose by providing habitat for indigenous peoples and allowing fish aquaculture. As a result, for fisheries in seasonal pond, dynamic zooplankton compositions are required. Copepod sensitivity to organic and inorganic contaminants in aquatic environment allow them to declare the pollution status of their respective habitats. And last one is my reference that I follow to conduct my study, and that's on from my side. Thank you, everyone. Thank you all.
Thank you, Mr. Islam. Are there any questions from anybody? So has this kind of a study on copepods been done, and are there any accepted levels for these or other bacteria for the kind of fish you're talking about, for fish aquaculture?
Thank you, sir, for your question. Sir, I just want to correlate with cyanobacteria and diatom with copepod, because according to other study, copepod is the most important sources, natural food sources for fishes.
Natural what? Say that again.
Natural food source.
Okay.
So I just selected some freshwater pond to see the copepod biomass and keep the aquaculture friendly.
Okay. Thank you. Are there any other questions? Anybody else? All right. If not, is that the last talk of this session?
Yes, sir.
Okay, then sorry, go ahead, Brushti.
So all the presentations for today's session are over. So I request Dr. Ruth Malik sir to please express his views about this session.
No, I really enjoyed it. It was quite a diverse session. And, it ranged from several talks in the area of biotechnology coming to a little more, basic life sciences area. And then we went on to behavioral science and Ayurveda, and we finally ended up in, having better conditions probably for growing fish. So I quite enjoyed this. It would be nice if for some of the specific talks, they were more observational, if the authors could give a little more insights into what is expected in the future or where these studies might go, that would have made the sessions even better. But still, it was quite satisfying, and I enjoyed the few hours that I've spent with you. Thank you very much.
Thank you, sir. Thank you so much for your kind support and cooperation, and thank you for giving your precious time to us. And we always hope to remain associated with you, sir. Thank you.
Sure, you're most welcome, and I hope to see you when I'm there in Bordi next time. Thank you.
You're most welcome, sir.
Yeah, bye. Yeah.
So I hereby conclude today's session, that is session A. So we'll meet tomorrow with another sessions.